book Mismeasuring Our Lives by Joseph E. Stiglitz, Amartya Sen, and Jean-Paul Fitoussi illustrates very well why we need accurate and reliable measurement systems. Not only in a social and economical aspects, as the book highlights, but also in all science and engineering disciplines. In order to do this, we often turn to optics and optical sensors to provide these crucial measurements. In this spirit, Cisma, in collaboration with the Milan OSA student chapter, the Young Minds, Scope, and FISUA, have put together this meeting to discuss about some of the state-of-the-art advances in the field of optical sensing and its future perspectives. And we would like to thank SUPA and the CDT-ISM for their financial support for this meeting. Professor Andy Harvey, experimental chair of experimental physics at the University of Glasgow and director of the Centre for Doctoral Training in Intelligent Sensing and Measurements, will start the day with his opening talk, Computational Sensing and Imaging, What's the Point? We will then hear 10 magnificent talks from researchers all around Europe and beyond, along with networking activities as detailed on the proceedings that we sent yesterday. Please let me remind you that the talks will be recorded, so be aware of that. Thank you all for attending the first edition of Eumos, and I hope you enjoyed the day. Now, Andy, over to you.
Sorry, I'm not quite sure what happened to my sound there. Anyway, thank you to Salva and to the organisers for inviting me to speak today. I think they've done a fantastic job in recent years of putting on a sequence of conferences, and this is just another fantastic example of a brilliant international programme, so I feel very honoured to be part of it. I'm going to talk very briefly about a recent immense body of research which is bringing computational processing within sensing and imaging systems. That's to say, the use of computational recovery is integral to the process of how we record an image. So this is an immense field, and I can only really touch on some of what's been achieved, but I'll give a little bit of a historical perspective, starting off with back in the 1960s, when we can trace back some of the earliest work. And in those days, we still needed a very sophisticated instrument and a little bit of computing that was available at the time to achieve something new. And then I'll quickly zoom forward to the 2020s and give you an example of where we've got to now, where we can have some very simple optics, but an awful lot of computing to achieve something very similar. I'll then spend some time talking about some recent work we've done in my research group, and this is concentrated on doing things that we couldn't possibly do without a computational imaging system. So hopefully at the end of that, I'll convince you that there is some point in bringing computation within the sensing and imaging. Let's get back to where we started at. So if we look back a long way to before the 1900s, of course, if you wanted to measure a spectrum, you have the technique that had been used by Newton to see to split up light into the colours of the rainbow. It's very simple. We have a light source and a prism, and we rotate the prism, and we can pick off wavelengths in time sequence. So it works, but it's very slow. Now, just before the 1900s, Michelson presented the Michelson interferometer. In that case, it was using coherent light, but it was then realised that if we put into that broadband light, white light, we record this interferogram, and mathematically it's easy to show that if we Fourier transform that interferogram, we can get the spectrum, exactly the same spectrum as we obtained here. However, given that Fourier transforms had to be done longhand, I guess in the 1800s that would have been with a pen and paper, that was purely a moot point. There was no way of calculating the spectrum. But in the middle of the 50s, it was shown by Jacquinot and Felget that if you could do that, then there was this enormous advantage that you could get, that the system was about 1,000 times optically more efficient. So it really would enable you to record spectra for the first time in areas of the spectrum where there were very few photons, such as in the infrared. However, we still didn't have a computation technique until in the 60s, Cooley and Tukey invented the fast Fourier transform. And as many of you probably know, that reduced the computation time from going with n squared, where n is the number of points, to n log n. So it massively shortened it. So that was an algorithm that was important, but computers were very much in their infancy until Moore's law took off in about the early 70s. And what that meant is we now had an efficient algorithm and we had efficient computers at low cost, and that enabled the launch of the first Fourier transform spectrometer. So the message we get here is that going from optical processing, the development of some novel algorithms, and the computational power that Moore's law gives us enables us to do things which would not otherwise be possible. But bear in mind, this still needed a very refined interferometer to produce those nice co-sinusoidal fringes. Let's wind the clock forward to this year, and we can see we've gone to another extreme. We can now use some very simple optics to form that pattern. For example, if we take a laser light and shine it into some disordered medium like a diffuser, we'll get a speckle pattern. And interestingly, we see that a very small change in wavelength will massively change that speckle pattern. So this speckle pattern now replaces the sinusoidal fringes that we get in the Fourier transform spectrometer, but we don't have a nice mathematical relationship like a Fourier transform to compute. We've got something much more complex. In effect, it's much more random and noise-like, and it requires something more like a lookup table. And there are many ways to tackle this. A very modern way is to use something like machine learning, where we take the input and we work out what inputs correspond to what wavelength distributions, and we can calculate that very easily. Now, the key difference here is we've gone from a very sophisticated Michelson interferometer, which would cost thousands of pounds to make something that works well, to a simple diffuser costing a few pence and a camera costing a few pounds, and some relatively sophisticated computation algorithms. But we can take this concept even further. We can use this for imaging objects based on the speckle that's produced, and importantly, we can get actually the phase of the object as well as the amplitude. And we can even use something like a bit of diffuser, for example, the plastic wrapping that you receive your Amazon parcels in. We can image through that, and we can actually, by calculating what the imaging process looks like, the so-called forward model, we can even reconstruct a 3D image in a single snapshot. So the key issue here, in comparison to the Fourier transform approach, we've now got to incredibly simple optics, but some very sophisticated computation to recover images, and you can see that there are lookup table-based approaches, and you can have various optimization processes that are quite slow but computation intensive. But the underpinning principle behind all of these computational sensing or imaging techniques is we have some optical processing which we develop, knowing that we can use some computational reconstruction which approximately compensates for the optical processing here to get an output that's a reasonable approximation to the object. And often we can do that in a snapshot because the optics does some sort of coding of information from the object onto the sensor. Why can't I get exactly the same output as my input? Well, that's because at some point there's noise. And that is the problem with all of these systems. In fact, it's also a problem for Fourier transform spectroscopy, that the inversion process, the computational reconstruction, is sensitive to noise. So we can see that the role of the optics now is to somehow encode information onto the sensor and make it robust to that noise. So this is where we've got to in the 2020s. This is an extreme example of very low-cost optics and sophisticated computation. I'm going to talk about some work we've been doing in my lab to try and get around the laws of diffraction. Now, the laws of diffraction are so important, they've been written in stone. This is Abbey's memorial in Jena in Germany, and there's the equation that says the smallest thing I can see is proportional to the wavelength divided by this thing here, which is the numerical aperture. That is something that's set by diffraction. The result of that is if I want to see something really small, I need to have a high numerical aperture, and the corollary of that is I have a very small depth of field. So if I want to see the features inside a cell, I can't simultaneously image many cells together. So that's a problem that diffraction causes. So I'm going to talk about some work we've done to get around that in enabling us to do microscopy in 3 dimensions in a single snapshot, and also to do gigapixel microscopy. 2 things which are fundamentally not possible given the simple laws of diffraction that are captured in Abbey's monument here. So probably the dawn of computational imaging was probably this paper here in around about 1995. This is imaging rather than sensing, but they were very closely related concepts. And built on a technique called synthetic aperture radar, Dowski and Cathey realized that if I put a special phase function, like a little sheet of glass in the lens here, I change an imaging system which goes to a point to one that goes to this sort of L shape here. So what is this L shape? This is like an optical encoding of the image. The optical encoding of a spectrum was done in the Fourier transform spectrometer, done with a sine wave. With my lensless imaging, it was done with this sort of speckle pattern. But within this computational imaging process I talk about here, is this special L shape. And this has a very good property. This enables a high contrast image that is insensitive to the addition of noise. So what can we do with that? Well, this shows in a conventional imaging system how we can have things that are in focus, but there's always something else that is out of focus. We can't image everything in focus simultaneously. But by using this special mask approach here, I can image everything in focus, or almost. There are some aberrations. Interestingly, in parallel with Dowski and Cathey's work, or rather some years earlier, Michael Berry showed that mathematically, at least, you could have an airy packet, so-called certain packets of described in a quantum mechanical sense, which move through this accelerating caustics, just like this optical wavefront does here. And they did this using a similar phase modulation. So that was actually 20 years before this demonstration by Dowski and Cathey. But it was another 10 years after this date, not until 2007, when these were first demonstrated as freely propagating beams optically. And this is the phase function here, which is put into the lens here. So, and these are the experimentally recorded point spread functions. The interesting thing is that you see these point spread functions move sideways, just as they follow this caustic here, and they're also a bit degraded. So in my lab, we were wondering, could we somehow use this increased depth of field and this lateral motion to record imaging in 3D and through a large depth of field? And this is how we achieved this. We used the imaging with that mask in place, and we applied the inverse function because we know what that L-shaped point spread function is. And this is just a simple pollen grain, always imaged in focus. But interestingly, in a single snapshot, we can also image it in 3 dimensions. And this is the same applied to a coin. This is a scratch in a coin imaged in 3 dimensions, and some biological samples and an integrated circuit. So in a snapshot, we can now image in microscopy in 3 dimensions. So we can do this in high speed. But a particularly attractive application is an even more simple implementation where you're imaging single points. Now, without computational imaging, you can imagine how you might do this. Here are some fairy lights. We can see that this fairy light here is in focus. This one's a little bit out of focus. It's come towards us, and this one's behind, and it gets a bit blurry. And if it goes a long way behind, I can see that we form a big disk. And the reason for that is that when I try and focus light from my fairy light to a point, if it's in focus, it forms a point. As it goes out of focus, it gets bigger and bigger. So in principle, if I simply measure the diameter of this spot, I can work out what its Z location is, how far away it is, and I could map out a 3-dimensional map of the fairy lights. But the problem is there's not very much light in these disks, so any noise means that I can't do it anymore. But that is solved by the use of this so-called wavefront coding, or the area encoding that I mentioned. And we find that this point spread function never loses its energy, propagates looking the same, but it curves a little bit. It's hard to measure the curve in one direction, but we invented a new type of phase mask here, which it produces this point spread function, which gets wider and wider with displacement. And it turns out we can recover that to always be able to record 3D location of points. So if we inject some fluorescent beads, for example, into a zebrafish, and then we can image those using this system here, these little L-shaped point spread functions as they move around the camera. And I can use these displacements of these point spread functions, the distortions, to work out what their location is in 3 dimensions. So as these point spread functions move around through the scene, I build up this 3D map of the blood flow within my zebrafish just by localization of the point spread functions. So this builds up over a period of time, and you can see the blood vessels building up here along the dorsal veins, along these veins here, vessels here, which go through the zebrafish's ribs. And if we compare that to what we achieve using a conventional 3D system, that's a light sheet, we can show that all the blood actually does go within these blood vessels. But we need this snapshot technique, which we can only achieve using computational imaging, to be able to reconstruct these at video rates. And we can even locate the pulsatile blood flow variation within the zebrafish's heart. We can also use these techniques, for example, to measure the forces within a cell, or indeed we can use this to image things which are much smaller than the diffraction limit. Normally that would limit you to about half a wavelength of light, but we're using this to be able to enable us to image biological structures which are perhaps only 20 nanometers apart. Now, the other thing that diffraction stops you from doing is being able to record very high pixel count images with very high spatial resolution. So I'm going to talk now about how we've used computational imaging to enable us to produce a gigapixel microscope, all for the cost of less than £100. This is the device we've made, and the key issue here is we have a special type of illumination, a little camera here, and we take this information off to a computer. So the microscope itself cost £100. The computation is much more sophisticated, and that's the quid pro quo. Simple microscope, complex image reconstruction. Oh, my apologies. I'll turn that sound down. This is a system we use. We use a very simple camera, and if we record a conventional image with this, we get something that's quite well focused in the middle, but is then very blurry. But our special technique involves switching these illumination lights on one by one and constructing lots and lots of images, and then we computationally reconstruct a very wide field of view image. In this case, it's 4 square millimeters at less than a nanometer resolution. Typically, this would require a microscope that would cost probably several tens of thousands of pounds, typically about £50,000. And we're able to do it with very simple optics for about £100. So how do we do this? Well, here's my sample, in this case not a biological sample, just a test target. We illuminate it with an LED at one position, and the lens that we use to form the image here records a spectrum which is blurry. That's to say, it's like a low-pass filtered image. But if we illuminate with another LED, we end up recording just the high spatial frequencies, so we get something that looks edge-enhanced. And that's one example of an edge-enhanced image. As I move this LED illumination around, I get many more such examples, and I build up the spectrum in time sequence. So I've got all the spectral information, but I can't really combine those together by adding intensities. I have to put this into a special algorithm which tries to use the physics of image formation to produce a self-consistent estimate of what that image probably looks like based on these many different measurements with different illuminations, and eventually reconstruct a very high resolution image from these multiple images. This is something that fundamentally cannot be achieved without the computer being built into the microscope. So of course that works, but it takes a long time to record all that data. So one of the things we did was to use a camera array to speed up the process. So we record many images simultaneously for each LED illumination, and that increases the speed by a factor of 10. But the computation gets much more complicated. Somehow, even though these cameras all record the data incoherently, we have to be able to add those coherently. And that's what that algorithm enables us to do that I showed you. But the consequence of this is that, well, we haven't yet produced a gigapixel image, but we've produced a 0.1 gigapixel image. So this is an example of this image. If we zoom into this, this is the raw image, and this is the high resolution image that we can reconstruct at various places across the scene, correcting for the aberrations that are evident in the optics. So very simple optics are highly aberrated, but we can correct for that with the algorithm. And we can also reconstruct the fade, not just the amplitude. And that high speed enables us to do things much more quickly, so we can also image moving cells. This is a rather old image now. We've done much better recently, but we haven't yet published that. So by using these computational techniques, we can do things which fundamentally aren't possible by the techniques, but also it gives us the option of doing things at a much lower cost. So this is just a couple of examples of what we've demonstrated in my lab, high-speed 3D and gigapixel microscopy. But we could also use similar techniques, and others have done this work to do snapshot spectral imaging, and there's a theme here about how we're trying to get what is effectively three-dimensional data through a two-dimensional detector, and that needs some optical processing. Other things you can do is you can produce cameras which are essentially flat. Normally we might think of the high magnification lens being long, but we can now make them flat by using multiple cameras, or indeed in Duke University in the United States, they've demonstrated gigapixel imaging, achieving the diffraction limit of a model-sized lens for the first time at gigapixel resolutions, or 10 gigapixel resolutions. This concept of encoding information into various patterns that also enables you to do things like put an image, in this case a letter 3, through a single optical fiber. So I can imagine putting this optical fiber inside my body, and it might only be a few tens of microns across, but I can send an image through that in a minimally invasive way. Another recent work is being able to do things which you think would be fundamentally impossible, and that's imaging round corners, and that can be done just by simply understanding the shadows cast on the ground here, or it could be used by some sort of time gating technique. And this is another development that's happening at the moment, is this transfer of these computational techniques to not only X, Y, Z wavelength, but also to time, to enable us to do fundamentally new things. So in the future, this is probably the future of computational imaging. We have computational imaging built into our mobile phones now, but more and more that it's enabling us to do new things, which are increasingly having impacts on everything from microscopy right through to biological imaging. So that was a rather quick excursion through this field. If you want to know a little bit more, then there's a really nice special issue on computational optical sensing, which you can see from early 2020. Thank you very much for your attention.
Okay. So next speaker will be Dr. Cristian Manzoni, who is a senior researcher at CNR IFN Milano, and he will be talking about hyperspectral imaging and microscopy. So please, Cristian. And for all the attendees, if you have any questions regarding the talks, please type the questions in the chat, and we will go through the questions at the end of the talk. Thank you.
Okay, thank you, Chiara. First of all, can you hear me and see me?
Yeah.
Okay, you can also see my presentation, I believe.
Yeah.
Okay, great. Okay, so first of all, I would like to thank you very much for the kind invitation to a very beautiful meeting. And already I found the first talk by Andy really inspiring, because it was very, very interesting. And actually part of what I will talk about will be connected to the first part of his presentation, actually. So the talk will be about hyperspectral imaging and microscopy. So the capability of measuring, let's say, an image, or like the capability of taking let's say, a spectrum from an image. So during my talk, I will first of all introduce the concept of spectral imaging. Then I will show you some applications. In particular, I will show you the applications that we, let's say, taken from some measurements in our labs directly. So let's see, first of all, very basically, the concept of spectral imaging. What's the meaning of the spectral imaging? So you may know that a spectrometer is, let's say, a device which enables to see which colors are inside a ray of light. Okay, so for example, if you take a light bulb, then by a prism, you can clearly see the spectrum which is produced by this light bulb. But actually, and so you can do that with a flame, with a light bulb, with the sun, with any source of light. But you can also do that if you take the, let's say, the light reflected by an object. In this case, actually, the light reflected by an object tells you or gives you, provides information about the object itself. Okay, so in this case, the red apple is already telling you if it will be tasty or not, for example. So red apples are my favorite, for example. So which kind of information can you get from the spectrum? Well, from the apple, that was the light reflected by an object. But actually, spectrum also may mean fluorescence. So in this case, if you excite some tap water or tonic water with ultraviolet light, well, in this case, you can see that the tonic water has a very strong fluorescence. So even though tap water and tonic water look the same under ordinary illumination, with ultraviolet illumination, fluorescence is different, which is telling us that fluorescence is also giving us information about the chemistry in some way of the object that we are observing. The same also happens, for example, for leaves in trees, where the color, which is changing, going from summer to fall and then winter, is telling us that something is happening inside the leaf. Okay, so for example, the amount of chlorophyll is related to the color of the leaf. And so this is telling us that measuring a spectrum will give us a lot of information about the object that we are observing. So actually, we can measure the spectrum of an object, which can be reflection, absorption, transmission, fluorescence, and also we will see Raman, for example. Well, we can measure the spectrum in principle at microscopic scale. This will be useful, for example, in the case of biology, where you can recognize tissues and cells, or about nanostructures, where you can also study the interaction of light with nanostructures. You can also measure the spectrum at macroscopic scale. So here, again, going back to the fruit, by looking at a group of objects, you can already spot point by point if there is something wrong with some of these objects. So in the case of food, for example, you can spot if there are some apples or some oranges which are not good. You can do also the same by looking, I don't know, for agriculture, for example, you can look at a tree and tell if there is something wrong, some disease, some parasites in the trees. And also you can apply, let's say you can measure the spectrum for an object also from very far away, so at mesoscopic scale. So in principle, by looking at the spectrum coming from an object which is very, very far away, you can, for example, from the surface of the Earth, you can monitor fires, water resources, soils, and so on and so forth. So now, the concept is which kind of information then I need in order to collect the spectrum from an object? Well, of course, the information is called the spectral hypercube, which is let's say a three-dimensional amount of information, because two dimensions are about the coordinates, the spatial coordinates of the object, and the third coordinate is the frequency or the wavelength. And so the stack of all this information is called the spectral hypercube. And once you have in your hand the spectral hypercube, then you can do many things. For example, you can extract an image at a single frequency and analyze what's happening at that frequency. Or you can, for example, extract you can sit on one pixel instead of one frequency. You sit on one pixel and you see the spectrum at that pixel. Okay, so the spectral hypercube can allow you already to get this very basic information. Also, with some computational tools, you can also extract a lot of other information about the chemicals, and also about if there are some points in the image which have the similar spectrum, then you can relate them, and so on and so forth. Now, the concept of spectral imaging is pretty complex because, here again, you have the ray of light that goes through a prism and then onto a screen. And then if you, instead of your screen, if you put an array of n sensors, this means that you are encoding your spectrum into n points, n coordinates. But now you can clearly see that you go from 0 dimension, which is a point, because it's a line, into 1 dimension, which is an array of sensors. So measuring the spectrum means increasing by 1 the dimensions. And so, of course, if you, as I was mentioning, if you're measuring the spectrum of an image, then in this case, you need actually something, so a sensor, let's say, in 3 dimensions. And here comes the problem. So how to measure a 3-dimensional information with typical sensors? And typical sensors are, for example, 2-dimensional sensors. Okay, so there are various approaches to do this. So here are, for example, the snapshots imaging approaches. This is pretty easy because simply you put a filter, you glue a filter or a matrix of filters in front of your camera, and then in one shot you can get information about all the colors. But as already you can see here from this picture, actually you're losing some spatial resolution in doing this, of course. The more bands you have, the smaller is the spatial resolution that you get. Otherwise, you can put many filters in front of the image. And in this case, then you acquire each image at each filter. Or otherwise, you couple a spectrometer with your imaging system. Again, but all these methods actually are the most famous one and also the ones which are commercially used. However, there is another one which was actually introduced in the previous talk by Andy. The other approach is the Fourier transform spectroscopy. So the concept is based on this idea. So you generate 2 replicas of light. Let's consider a ray of light. 2 replicas of light. Then you go into a detector, you record the energy as a function of the delay between the 2 replicas of light, you get the interferogram, and then once you get the interferogram, then you do the Fourier transform here, and then you get the spectrum. So you get a spectrometer without a spectrometer. There is a lot of computation here, as it was previously mentioned, but the idea is that you do not need to disperse your light, you do not need to filter your light, which is typical of bandpass filters, typical of spectrometers, typical of gratings. You do not need to filter out the light. And so you can get a spectrometer without a spectrometer, without a dispersive element. And the advantage of this technique is, well, first of all, the throughput, because you do not need to filter the light. And indeed, one of the very first examples of the Fourier transform spectroscopy was applied in astronomy, where typically you have very few photons to measure. And indeed, it was used in the 60s to measure the spectrum from Orion Nebula. There is another advantage, which is not only the throughput, but the fact that you can parallelize these methods. Because instead of measuring a ray of light, you can measure an image, like this one, for example. And so in this case, if you measure an image, then you can record a set of many images as a function of the delay. And then you can do pixel by pixel the Fourier transform. Okay, so this is giving you the possibility to measure a spectrum by generating many let's say, images at different delays and then performing the Fourier transform. Now, the problem, which was also mentioned before, is that you need, let's say, to adjust the delay with a precision which is much better than 1/100th of the optical cycle. Okay, so you need a very high precision in generating this delay. So one way to generate this delay is by the Michelson interferometer, which, so the very first version of this was done in 1881 by Michelson, who built the Bayer interferometer, and actually he failed. And the reason why he failed was that he was not considering the perturbation, the effect of the perturbations over this interferometer. Because actually he, in order to do Fourier transform spectroscopy, or also in order to build the proper interferometer, you need to have super stable mechanical equipment. So any perturbation on one mirror then destroys the delay. And I would like to remind you that we need to be precise at the order of 1/100th of the optical cycle. So in the visible, it means better than 3 nanometers, or let's say 6 nanometers if we consider the total delay, 3 nanometers if you consider the movement of a mirror like this. And indeed, Michelson could repeat the experiments in 1887 with the help of Morley, because he built the interferometer over a stone, which was very heavy, floating in a pool of mercury. So he had to isolate the interferometer from the rest of the world. That's the reason why the Fourier transfer spectroscopy is not very common in commercial devices in order to do hyperspectral imaging. And so here I would like to briefly introduce actually another approach to build an interferometer very quickly, which is the one that was developed in our labs, and later I will show you the results exactly from this device. So this device is based on birefringence. So if you go through a birefringent crystal, the result is that you duplicate your image. You generate 2 replicas because the propagation depends on the on polarization. In this image here, you can clearly see that the one line becomes 2 lines. So I'm generating 2 spatially separated replicas. But actually, if I rotate, if I let's say cut the crystal properly, I could also generate 2 not spatially separated replicas, but 2 temporally separated replicas, as shown here in this panel here. I have one, well, here let's consider a pulse, because it's also working with laser pulses. Here I have a ray of light, a pulse, which goes through a birefringent crystal. If it is suitably cut, then I can generate 2 replicas which are delayed. And I can tell you that this is a super stable interferometer. Okay, nothing will be able to change this delay. Only I need to change the thickness, okay? And in order to perform the Fourier transform spectroscopy, actually I need to change the delays. And so this interferometer is beautiful, but it's not working at all because I need then to change, to tune, to modify tau, okay? And to record the intensity as a function of tau. So actually what we did, I'm not going to enter into the details, but it's we used twice the birefringence. So one plate here to generate 2 replicas, and then another plate to change their delay. And this plate is cut so that by moving one of the 2 fractions of this plate here, I am able to change the thickness. Since delay is proportional to the thickness, we instead of moving a mirror, we move a crystal in order to change the thickness. And what advantage of this interferometer? Now, this is exactly acting as a Michelson interferometer, exactly. However, it's much smaller and way more robust, because as you can see here, it's 4 centimeters, so it can stay on your hand, actually, and it's extremely robust. And so let's see how this interferometer can be used, can be applied to Fourier transform spectroscopy. And so let me show you some examples on how Fourier transfer plus this interferometer enables you to do hyperspectral imaging, so to record images with also spectral information. This is the very simple device. So interferometer, very compact, can stay in your hand, plus a commercial camera, a CMOS camera in this case, plus a camera lens. Very simple device. And let's see what we could do. So the first thing that we did was to measure reflectance. Okay, so the reflectivity from an object. And this was done in collaboration with the cultural heritage group of our department. And so we took an image of this reflectivity image. So we illuminated with white light and then measured the reflectivity from this very ancient cartonnage. This is in principle from 2,000 to 3,000 years old. And so we could measure the reflected light, and we could, you can see here, these solid lines are actually the reflectance from the object. Okay, so the reflected light from the object. And from the reflectance, then I could synthetically generate actually the colors of this cartonnage. We could do also another thing, because in the beginning I was mentioning, okay, from the object we receive the reflected light, but also we receive fluorescence from the object. Then indeed, by illuminating with a red light, with a red LED, we could also measure the fluorescence. And first of all, we could measure the spectrum from each point of the image. And so this is the spectrum of the fluorescent dye in this cartonnage. We could identify a dye which is called the Egyptian blue. It was the Egyptian blue was really the very first artificial dye which was made by mankind. And as you can see here, it's spotted in green. We could clearly see where the Egyptian blue was used in this picture. It was not everywhere. Okay, so this is one example: identification of the pigments and also localization of the pigments. Another very interesting application actually is the following. Here we took also an image of an artistic glass window. This is from a church. This picture here was taken with a commercial Nikon camera. Okay, this is not hyperspectral imaging. This is ordinary imaging. And then, why did we took an image here of glass? Well, because it's you know that the colors from the glass come from the nanoparticles, which have a quite sharp spectrum, so less boring than reflective spectrum. Okay, more interesting probably. So we took an image of this glass window. We went into this church and took an image, and in particular, we took an image of this detail here from San Giacomo. And here is already the result. So we took the image, we did the Fourier transform, and here I'm showing you some images from the, let's say, at specific wavelengths. But please remember, this is after taking the Fourier transform. Then I have the whole hypercube. And then from this whole hypercube, I am extracting the information from one wavelength. And as you can see here, here, for example, I gave you 6 examples, 6 wavelengths, and they are all different from one with respect to the other. So actually, they are each wavelength is carrying some specific information. This is the synthesized image. So from the spectral information, I'm able to reconstruct the color image, which is very similar to the one taken with a commercial Nikon camera. But now I have information over a potentially infinite number of wavelengths. And this is, if you want, the synthesized image. Well, sorry, the sliced image. So I'm showing you what would like the image at each specific wavelength. Okay, so I'm showing you let's say each wavelength, and so you can clearly see what's happening at each specific wavelength. So any element appears at any color. This is due to the very narrow bandwidth of the transmitted light from the window. And we could do something more, for example, because we could focus on the blue tiles. Okay, here we have many blue tiles in the picture. If you know the spectrum, you can also use some codes. In this case, it was the automatic k-means segmentation. And you can use these codes, for example, to classify, to cluster your pixels. And so, for example, we saw that in this window, there were at least 5 types of blue tiles, which we could spot because the automatic k-means segmentation could say, okay, look, I found 6 different spectra in the blue. And we could also identify a fake one, because the one which we colored in orange, which is sitting here and here, has been probably changed, because as you can see, it's not transmitting in the infrared, which means that probably at a certain point it broke, and someone else built the blue tiles, which was then replacing the broken ones. Okay, so it's also a way to discover if something changed, or if also there is some fake elements here, for example. Of course, the camera and imaging can be used also from remote. So this is our camera, and we are taking a picture from a lake and an island on a lake. And here are the spectra in different points. So the trees, here, for example, the water of the lake, and so on. And so here you can clearly see the two peaks at, let's say, around 800 and 850 of the two peaks of chlorophyll, for example, which is telling us that the trees are true. Okay, they are not plastic, they are not fake, there is no camouflage, and so on. And so in the middle, you have the reconstructed image, and here again, the sliced image at different colors. And you can clearly see that the image changes contrast when we go from the infrared. So here it's the infrared, where the trees are reflecting, to the visible, where they are absorbing. And so you can clearly see that when we switch to the infrared to the visible, I clearly change the contrast completely. As a last example, I would like to show you the application of imaging to microscopy. Okay, so the concept is pretty simple, because we put the interferometer into a commercial microscope, and in this case, for example, we imaged some cells which were stained by Alexa, which are sort of some dyes, actually. So we could clearly identify the nuclei and the membrane of the cells. Okay, this was a measurement which was done in collaboration with Marco Ereni from Humanitas Hospital. And so we could clearly demonstrate that by the fluorescence, in this case it's fluorescence, by fluorescence spectra, you can clearly see different parts of the cell. Also, using fluorescence, you can, for example, use this not only for biology, but also for material science. Here, for example, I show you a microscope image of a mixture of cadmium orange and Egyptian blue, which are powders, whitish powders, actually, and which were mixed. So one way to identify them is simply by taking a fluorescence image. So here you can see that by analyzing this image, we could see that in this pixel here, the spectrum is for the cadmium orange. And in this point here, we had a pixel fluorescence from Egyptian blue. So we could clearly spot the two elements, or the two, let's say, constituents of our sample by looking at the fluorescence. And so here, for example, again, you see the sliced image. Okay, so you can clearly see who is where. And this is the synthesized image. So one very last example. So don't trust the number here of the slides. For some reasons, here it tells 37. It's not true. Okay, don't trust it. So I'm going to very close to the conclusion, because I have shown you that we can measure reflectance or transmission in the case of glass. We can measure fluorescence in the case of cartonnage, in the case of cells, in the case of Egyptian blue and cadmium orange, for example. But we can also measure Raman. You know that the efficiency of Raman is extremely small. Okay, so but luckily we have now our high throughput interferometer enables to collect all the photons coming from a Raman signal. And so we could collect, now this is a very preliminary result, we could collect this typical image of polystyrene and PMMA beads. And each of them is colored in green and yellow according to their Raman spectrum. So we could chemically also identify the object by taking this image. Now, please note that this image here is 0.25 million pixels. Okay, the acquisition time is extremely short thanks to the high throughput of the Fourier transform approach. 36 minutes, which means that it is as though you are sitting. So typically Raman spectra or Raman images like these are acquired in a totally different way. It's not used by using Fourier transform, it's not by using wide field microscopy, but sitting at each point. You sit at one point, you measure the spectrum, then you go to the next one, and next one. The integration time is called the dwell time, and actually let's say typically the dwell time is of the order of tens of milliseconds. or hundreds of milliseconds or seconds. In our case, we could acquire this image in 36 minutes, which is equivalent to a dwell time, equivalent to, because we were not using raster scanning, but wide field imaging, but it's equivalent to 8.6 milliseconds, which is very promising result. So just to conclude, and just to give you a brief summary of this presentation, so I have introduced very quickly and briefly the concepts of spectral imaging. Then I focused a little bit more about Fourier transform spectroscopy. And then I have shown you a couple of examples of remote sensing in which the object is macroscopic but far from me, and microscopy in which I can look the details of a sample, which could be cells, tissues, material science, for example, and also I could measure Raman. So with this, I would like to conclude my presentation, acknowledging all the people who took part to this large amount of experiments, and also the founding agencies actually which enabled us to do this kind of experiments. And so with these, I really would like to thank you for your attention.
Thank you very much, Cristian, for the very inspiring and exciting talk. So we have a couple of questions for you. We can go through all the questions, so please, if you can type the answers later when we finish the
Sure. Okay. Yes, definitely.
Okay, so
yes.
I'll go with the first one. The first one is from Sean Bomer. So he asks, for your twin setup, you appear to have an air gap between the two wedges. Does this have any effect on the beam alignment of the system, as it seems one half of the beam profile has to propagate through slightly more material?
Yeah, this is a technical question, but it's totally true that you have to be very careful about this. So you can't open a big gap. In our case, we set up the experiment in order to have a gap of, let's say, 1 millimeter. If you do better, you can do 0.5 millimeter, and this is pretty fine. This is doable, of course, as you know, mechanically speaking, you can do that. So in principle, you should not open big air gaps. This is totally true.
Okay, and maybe I'll pick up another questions from Michele. Michele Lacerenza says, I'm interested in the application of spectroscopy to agriculture and food sector. I see that usually those spectroscopy devices lack of precision and accuracy. Do you think that the state-of-the-art research has got some problems, interesting solutions to that problems?
Well, well, of course that in agriculture it's not easy actually to be very precise in analyzing these let's say the spectral information and so on. But in my opinion now, and this is also a direction that we are taking, we would like to go deeper and better into the let's say agriculture. What we would like to see is also to apply very sophisticated and complex methods for analyzing the spectra in order to get better information. But this is something that we are going to, this is an activity that we are just starting, okay? But I feel that once you have the spectrum, and the spectrum could be either fluorescence from, for example, the chlorophylls, or simply the reflectivity, then you can get information. Of course, then there are some problems, because it's, you know, when you do imaging in agriculture, you are not in a lab. So you are not in the safe and easy and controlled conditions of a lab, where you have your sample, which is in front of you, you can control the illumination and so on. Of course, in agriculture, there are many problems which are related to the illumination, okay? However, I believe that once you have the spectrum, there are ways in order to be able to, let's say, correct for the illumination. I already found many interesting talks during the Photonics West conference, in which they showed that actually you can deduce also some information about the illumination. You can compensate for that, and also then you can deduce many really the reflectivity of the object. And this is possible not because you are taking an image of one point, but you are taking the image of the whole picture. And so by correlating pixel by pixel, you can get a lot of information. But there is a lot of let's say, processing in that.
Okay, thank you, Cristian, for the answers. So you can type answers to the next questions in the chat.
Okay,
So
thank you very much.
Thank you again. So we'll move on to the next speaker.
Hello, sorry about that. Can you hear me?
Yeah.
Can I hear you? Sorry, one more time. Can you hear me?
Yeah, I can hear you.
Brilliant, thank you. That was my bad. Sorry for the slight delay there. I'll be chairing this portion of the event. I'm assuming we're on to our first speaker, Gianmaria Calisesi. Please feel free to correct any horrible pronunciations of your names in any of these talks.
Yeah, sure. I know it's pretty difficult. It's Gianmaria anyway, but it's the same.
Cool.
Yeah, it's.
So you're a third-year PhD student at the Polytechnic University of Milan, and you'll be discussing some of your work on compressive sensing and light sheet fluorescence microscopy. Cool. Well, if you're happy to continue on, please do.
Okay. Well, thank you again for the opportunity of presenting here. And today my speech is going to be on light sheet fluorescence microscopy and on compressive acquisition protocols in this technique. So, okay, it's not working. It's not moving on. Okay, nice. So a brief outline of my talk. First, I'm going to go through the motivation beyond my work, together with some theoretical principle of light sheet fluorescence microscopy and compressive sensing theory, which is the compressive paradigm that I've used in my work. Then I'm going to show you how I applied them together in spatial modulation, selective volume illumination microscopy, how I try to achieve a volume as wide as possible. And finally, I'm going to go through some conclusion and future perspective of my work. So when dealing with living samples, or more in general, biological sample, one wants to have a reduced light dose, so to prevent the sample from suffer from any damage, any photo damage or any photo-induced related damage, together with a motionless scanning activity, so to prevent the sample from any further shock given by movements. Altogether, with these two, one wants also to avoid the formation of artifacts which may be a fingerprint of the microscopy technique that one can use. And altogether, also finally wants to obtain a large volume with the smallest degree of resolution possible. So with this in mind, let me go through the principle of light sheet fluorescence microscopy. So light sheet fluorescence microscopy is a microscopy technique in which an incoming collimated laser beam is tightly focused along one direction thanks to cylindrical lens, at least in its easiest implementation. And so this tiny light sheet excites fluorescence over a single plane. And then we have a wide field detection, which collects the fluorescence signal on a path which is orthogonal to that of the light sheet. So with this design, with this particular design, what we obtain is an intrinsic optical sectioning, since we're just shining light over one plane at a time. We also obtain an intrinsic higher speed and low photobleaching compared to point scanning technique, since we don't have to go through the every point of the plane. We have one plane at a time, and we also obtain an highest efficiency of the photon harvesting, since we are actually collecting fluorescence signal coming from every portion of the sample that we are illuminating light on, which is different from, for example, a confocal microscope, in which we are shining light over a cone of the sample, whereas we are just acquiring the signal from the in-focus points, so from just one point. So with this particular setup, we obtain a lateral resolution and an axial resolution which are given by the detection and the illumination objective numerical aperture, respectively. In particular, the axial resolution is determined by the light sheet thickness, which is again determined by the numerical aperture of the illumination. So in practice, what is done is that we actually infer volumetric information of the sample as it displayed in the video by optically cutting the sample. Of course, in microscopy, we don't do that on chocolate bars or on candies, but we do this in, for example, biology. And due to the fact that we obtain bleaching and intrinsic high speed, this approach actually holds a gentle microscopy approach to study in vivo dynamics, like development of embryos, for example, zebrafish embryos, which is tracked and followed over time. And here you can see how the mixed information between a bright field microscope and a light sheet microscope actually enables us to track the movement of the blood cells within the development of the embryo of the zebrafish. So volumetric scan in light sheet is in its easiest implementation obtained thanks to a motorized stage which moves the samples across the light sheet. So the light sheet is in focus compared to the sensor, and we just move the sample across the light sheet. But this can imply that actually the living sample may suffer from mechanical shock given by this movement. So in practice, what I want to obtain with my work is to achieve a motionless scanning activity thanks to adaptive elements or moving elements like galvanometric mirrors, electrotunable lenses, and spatial light modulators. So once again, in light sheet, we have the tightly focused of light over just one plane. But what I thought to do is that maybe we can go over this and we can extend this by not modulating just one plane, but by modulating more than one plane of different thickness over the depth of field of the detection objective, as displayed here. So this could, in principle, enable me to code more information in just one frame. And by keeping the orthogonality between the illumination and the detection, what I can do is thinking of integrating line by line what the sensor perceives, and to encode again some more information. And doing this with particle illumination could be achieved by, for example, a simple light modulator and some collection and condensing optics, so not compromising the cost of the overall setup by this much. And by patterning light, what I could obtain is to obtain the possibility to implement some compressed sensing or some compressed acquisition protocol, where compressed sensing has many application fields ranging from mathematics, from material science and biomedical imaging, and one of its most successful implementation maybe is magnetic resonance imaging. Well, but yet again, it has been extensively tested on in vivo setup, like imaging of brain of a baboon, and so it's a quite well-established technique. But more into details, compressive sensing is a signal protocol reconstruction technique that aims at reconstructing a signal starting from fewer samples, fewer measurements than those provided by the Nyquist-Shannon criterion. And in particular, this is obtained by solving an underdetermined problem. So let me divide the compressive sensing routine into three major steps. The first one is representation. So we want to reconstruct the vector C, which can be intended as any wanted signal. We can think of it as represented by a matrix W into another basis. W could be, for example, a wavelet transform, a Fourier transform, a Noislet transform, or whatever transform, a linear map that maps our former vector C into another vector X, which can be intended as to be sparse in the destination domain. Once we obtain the represented sparse vector, we can think of the measurement process in this space to be matrix multiplication in practice, namely the measurement matrix here is depicted as A, and the while the measurement vector is depicted as Y. So measuring, it just means having a multiplication, a matrix product between A and the sparsest vector X, which is nothing more than a representation of C. So once we have measured, here we see just that X show only few non-zero entries because we actually want this to be as sparse as possible. Once we have these entries of the measurement vector Y, we can think of recovering the vector X by actually optimizing for the sparsest solution possible. And this actually counteracts the ill-posedness of the problem. So in practice, in the easiest case in which the signal is already sparse in its former domain, the representation matrix can be intended as the identity matrix. So actually what we want to obtain is the sparsest possible vector in the spatial domain. And the spatial constraint is nothing more than, mathematically speaking, that an L1 norm, which is just the sum of the absolute value of components of the unknown vector. Okay, so by doing this, we can solve with starting from an ensemble of measurement which has fewer entries than those provided by Nyquist-Shannon criterion. But the elemental setup that I designed in order to achieve this had an LED source whose light is collected by a standard color illuminator design, which shines light over a special light modulator, which is called digital micromirror device, which modulates light. Then light is collected and refocused by a system, here depicted by two objective lenses, and refocused over the sample. Here, a digital micromirror device is nothing more than a matrix of tiny active elements, tiny pixels of 7 micrometer pitch, and each of these micromirrors can tilt along its diagonal according to any wanted patterns. Actually, this behaves like a screen, like a monitor, and so we can project any wanted pattern. In principle, this could be also non-binary, since we can implement dithering of these micromirrors over 2 levels. But in practice, this could lead to a decrease of the contrast of the pattern itself. So in this work, what I decided to implement is just binary patterns, so to obtain the highest modulation contrast possible. So this is the DMD screen projected over with the projected patterns. So I decided to implement a Walsh-Hadamard basis as patterns, as measurement patterns, since these bases actually have been proved to be highly incoherent. So have been proved to be highly to seize the information, the spatial information of the scenery in one of the most efficient way possible, together with Fourier basis or wavelet basis or noise basis. And so I'm just modulating not the whole DMD, but just a part of it because of the small depth of field of detection. And here I'm just modulating with different Hadamard bases, which are just the rows of the Hadamard family, which are just displayed here. And the overall acquisition at a given sample is displayed here. We just perceive the line integral over the different patterns, and we see how the scenery is flickering, so we have oscillation, and in this oscillation actually hides the information that we want to obtain, which is of course the volumetric distribution of the fluorophore. So I tested my technique at the first stage on fluorescent beads and on zebrafishes, which were differently labeled in the muscles, in the blood vessels, and even non-labeled. So with beads, what I wanted to obtain was an assessment over the optical feature of the setup, namely axial and lateral resolution. And here we can see how actually the technique yields a small volume of modulation, since we suffer from both the limited depth of field along Z, and on the same extent, we also suffer from the limited persistent length, which is given by the LED illumination. So we only have the central part of the volume, which is of use to the reconstruction. And in this region, which is quite elongated along Y, we obtain diffraction limited value for these two resolutions. So with this, with no extension, we obtain a volume of 200 microns along X, which is given by the persistent length of the LED, and of 120 microns along Z, which is given by the depth of field of the detection. And on Y, we are just limited by the sensor size, so we have 3.2 millimeters. And for this reason, I decided to go further on the investigation of this technique on elongated samples like zebrafishes. In this slide, what I wanted to show is how this technique compares to light-sheet fluorescent microscopy. And in terms of optical sectioning, these two are equivalent. Except for the fact that I have light dose shining the sample, which is 1 order of magnitude lower than that of light sheet microscopy with comparable exposure time. So I moved on, and well, why did I use an LED illumination then, since we do not have this improvement? Well, one fingerprint of light sheet is that we have shadowing artifacts given by the presence of obstacles like pigments on the skin of the fish, for example, which may hinder the propagation of light, which show high coherence. So by absorbing and by the presence of the absorbing center, we have a degradation of the quality of the light sheet itself. So by using a lower coherence, a lower spatial coherence source, what I obtain is that light can propagate beyond these centers, so that I do not see anymore this kind of striping or shadowing artifacts, which are a fingerprint of the light sheet technique. And this is why I choose an LED source. So I decided to investigate and to assess for a compressed protocols. So from the left to the right panel, we see how the compression factor is increasing. And here for C equal 2, I'm just using the 50% of the initial patterns. C equal 4 is 25 and C equal 8 is just 12.5% of the initial set of measurements. And we see how the overall, overall, the amount of information carried by the measurements is preserved, since we have, some details preserved. But if we zoom in, we can see how actually the tiniest information along the axial direction are not preserved, as in the case of this crossing of vessels, which actually is lost as compression is increased. But yet, I decided again to extend the analysis over larger samples. So here you can see a video of the acquisition of modulated patterns over an adult zebrafish brain. The modulation still is present, so misregarding the dimension, the increasing dimension, and yet the information which hides in the oscillation given by the different patterns, the different modulation, is still present. So this was a good test mark for my technique. Actually, the reconstruction, the volumetric reconstruction, is good on the modulated region. So with this, I decided to move forward. And what I demonstrated up to now is that pattern illumination together with an LED source could be implemented to obtain information which are equivalent to those provided by light sheet fluorescent microscopy. And I also shown and demonstrated how this acquisition protocol, differently from light sheet protocols, can be used in compressed sensing acquisition, so to further decrease the overall amount of light or the exposure time to use in our microscopy. So, but yet something could be modified. For example, a more efficient light collection could be of use to reduce, again, the exposure time. And in order to acquire larger samples, I could increase both the illumination persistent length and the detection depth of field, together with some optimization on the algorithm beyond compression, or even the convolution may be possible. So in the second part, I'm going to show this implementation in my setup. In particular, I optimized the illumination scheme thanks to a light pipe and a total internal reflection prism, which optimizes for the optical coupling of the incoming LED radiation with the DMD screen. I got rid of one of the two objective lenses, and then what I thought to do in order to increase the extension of the illuminated volume could be, for example, moving the illumination objective. And this was also achievable thanks to the fact that we can, thanks to a custom provided acquisition mode of the camera, we can read the sensor, the camera sensor, on a line-wise mode, so that we can synchronize the movement of the illumination lens with the reading of the sensor, so that the line that we are reading and acquiring is perfectly in focus with respect to the illumination scheme. So by this is quite different compared to what I've shown you before. That was the situation that I showed you previously, which resulted in a volume which was elongated, and it was really suitable for the imaging of narrow sample like zebrafish's embryo, whereas this translation situation is such that the extended volume is only limited by the sensor size, and we can think of acquiring samples which are of millimeters size, and for example, this could be the case of mouse brains. So I investigated a complete reconstruction over mouse brains. So I'm showing you the reconstruction, the complete reconstruction of the structure, the blood vessel structures in a mouse brain labeled. We can see how the tiniest details are preserved over a large volume. But yet, what we see is that, yeah, okay, isometric resolution is obtained along axial and lateral direction, but even if I got rid of the limitation of the illumination extension, we still see how the modulation within the depth of field suffer from the limited depth of field again, and so we can see some defocus artifacts at the border of our region of interest. But yet the blood vessel structure can be reconstructed over the whole volume, narrow volume, but yeah. So I decided to move forward and to extend the volume of imaging up to a factor of 7 millimeters. And this is again a whole mouse brain imaged, and we can see how it still suffers from the focus at the border of the region. But yet in the central part, we still have a great detail. Yeah, so I decided to move forward and to image different kind of sample in which neurons were tagged. So we have this pointy structure, and by inverting the problem, what I obtained, and to reslice, a tomographic reslice of the sample, what I seen, okay, is that actually my technique yielded some artifacts which were something very strange at the very beginning, but I discovered that these strange artifacts here were given by the photo bleaching effect of the technique, which might be given by the clearing protocol. And this photo bleaching actually acted as a coupling effect between modulation and time, which is something that we don't want to have. So in practice, what I see is that aside from the modulation effect of the different pattern, we also have a further modulation given by the photo bleach. And so this actually caused these kind of artifacts by coupling the spatial depth information with the time information. So by compensating for this exponential decay, I was able to remove these artifacts. So for instance, my technique was free of any artifacts, okay? And as you can see here from a tomographic reslice. So, but yet again, my technique on X and Y is not limited by anything, is just limited by the sensor size. Whereas on the Z direction, this is limited by the depth of field of the detection. So my final attempt to remove this limitation is to go for the convolution algorithm. So namely, image is formed, can be thought of as a convolution between the object spatial distribution with a function which is called point spread function, which is a fingerprint of the technique of the microscope. So we can think of this convolution in the Fourier domain. This is just a multiplication between the object spectral representation and the optical transfer function of the microscope, which is just the point spread function Fourier transform. And we can think of inverting this formula and to anti-transform to retrieve for the object distribution in the volume. So in my setup, the problem can be reformulated quite differently. In particular, we have the illumination distribution that is actually modulated by vector pattern P. So in practice, we have the illumination, which is the convolution of this pattern with the point spread function of the illumination objective. Then we have the object, which is just the volumetric distribution of the fluorophore X. So the overall image is given by the scalar product between the illumination distribution and the object distribution, convolved, as I showed you before, with the point spread function of the detection objective. So with this image formation, I implemented a Richardson-Lucy algorithm, which aims at, by knowing the point spread function of the system, this algorithm aims at retrieving the volumetric spatial information without the blurring effect given by the depth of field of the detection. So here you can see the deconvolved and just the inverted same slice 40 microns away from the focus. And Richardson-Lucy actually acts more like a denoise algorithm here, and as you can see by the comparison of the two. But yet Richardson-Lucy performed best at the very center of the volume. Here we can see how actually the denoise gets stronger and stronger, and it also displays a symmetric characteristic over the focal point as 40 microns away in the other direction. So with this, I just showed how multicolor light sheet microscope for volumetric acquisition of millimeters large sample can be obtained without the limitation given by artifacts of light sheet microscopy. And with this, I have also shown you how this can be coupled with compress and the convolution algorithm to enlarge the volume of interest. So with this, my presentation is finished, and thank you again for the opportunity, and I'm glad to take any question that you might have. So thank you.
That was a fantastic talk. I imagine your supervisor is very happy to have you as your PhD student.
Thank you.
It doesn't seem to, oh, there seems to be some questions coming in now.
Okay, let me check for them. Actually, my supervisor is not as happy as you may expect, but okay, maybe I should.
There is one here from Lorenzo saying, I wanted to ask you, how long does it take using a single sheet of light to illuminate the entire sample? And connected to this question, exploiting the multi-sheet analysis, what is the maximum fitness that would be possible to illuminate simultaneously? Quite an involved question.
Okay, so okay, yeah, let me start with one of them. Well, actually, I'm not, Lorenzo, I'm answering to the first one. I'm not using a single light sheet of light. I'm using multiple light sheets, but actually my technique has just some acquisition time, which is a factor of 4 or 5 longer than those given by light sheet. So typical values of light sheet exposure, well, of course, it depends on the fluorophores implied, but best case scenario, this might take for, let's say, 10 milliseconds to happen for the acquisition of a single frame. In my technique, it might take, let's say, 50 milliseconds, so a factor of 5. So the time scale are comparable, let me say. But yeah, I know I compensate for this longer acquisition time by shining the light, shining the sample with a smaller light dose. Let me say that. So okay. So second question, in your setup, you use a special light modulator, okay? Acquiring time. Actually, the main limiting factor of my technique, of my acquisition time, is the fluorophore itself, since most of the time I have sufficient power to go also into, I don't know, tens of milliseconds acquisition, but instead the fluorophore is not as efficient in terms of quantum yields. So it's not, the DMD is not as inefficient as one might think. So, well, of course, it acts as a diffraction grating, and this is the case of laser sources. By the low coherence of an LED, actually play on our advantage in terms of refocusing light. So, I mean, I would say that the most limiting factor is just the fluorophore itself. So, Federico, if I answer your question. Okay, does this technique is suited for fast? Well, it's not as fast as for fast phenomena, okay? So it's just for, I would say, hours time scale, like the one that I showed at the very beginning.
Thank you for answering those questions.
It's a
There's a
pleasure.
chance that some more might appear in the chat, so later on during the other talks, so if you can maybe keep an eye on the chat window for a little while, that would be appreciated. I think it makes sense to move on to the next talk so that my delay doesn't eat into our lunch break.
Hello everyone, and welcome back from your lunch break. My name is Greg Miles. I'm a PhD student at the University of Edinburgh, and I'll be hosting the next session of our European meeting on optical sensors. This session will be mostly focused on optomedical sensing. So there'll be 3 half-hour talks followed by a quiz, and I'll give you details on that shortly. And just a reminder that if you have any questions during or after any of the talks, please either type them out or use the raise hand feature at the bottom of the screen. So allow me to introduce our first speaker, Dr. Angelo Sampaolo. He's an assistant professor at the Politecnico di Bari in Italy and an associate researcher at the Institute of Laser Spectroscopy at Shaanxi University in Taiyuan, China. His recent research is focused on trace gas sensing, and his talk today is titled Tuning Forks in Gas Spectroscopy, How to Listen to the Music of Molecules. So if you're ready, Angelo, you can take it away.
Yes. Thank you for the introduction. Let me share my screen. All right. Okay. Do you see my slide?
Yes, we do.
All right. Okay, let's get it started. Good afternoon, everyone. I'm Angelo Sampaolo, and I work in Politecnico di Bari. I also do my research within the Joint Research Lab Polysense, which is a joint research lab between Polytechnico di Bari and Thorlabs. And today I will give you a talk about tuning forks, the role of tuning forks in gas spectroscopy. In particular, how to hear the sound of molecules. Okay, let me start from the most important branches of gas sensing application. One of the most important application for gas sensing is for sure the industrial process control. So basically, you may have the need to monitor leaks in oil and gas, or you may have the need to analyze the natural gas in order to prevent, contamination of the seabed, and also to predict some outcome of the drilling. Monitoring of combustion process, as well as chemical manufacturing and waste disposal. For what concerns the environmental monitoring, of course, the air pollution monitoring, as well as the detection of toxic gases, and of course, detection of early fires. And then, of course, we have the health and life sciences. In particular, most of applications are focused on breath analysis and on detection and monitoring of biomarkers. And of course, the security and safety, in particular, the monitoring of hotspot areas, just like airports, in order to prevent any kind of attack or possible terroristic attack, for example. Okay, let's focus our attention on spectroscopic techniques. And the first question is why they are different from, for example, mass spectrometry or chemoluminescence or gas chromatography. As you may have seen from the previous presentation, the spectroscopic technique relies on laser light absorption. So basically, we use laser sources, which are characterized by very, very peculiar characteristics of spatial and time coherence, and also super high directionality. We use this light with very precise energy in order to excite the transition, vibrational or rotational transition of the molecules. Okay, from the near infrared up to terahertz range. What are we talking about? We are talking about the degrees of freedom of the molecules. So basically, we are talking about stretching, bending, twisting, and stuff. So we know that this spectroscopic technique, the main features of the spectroscopic technique are the high sensitivity and selectivity, and the sensor that you can build by relying on these techniques are robust, portable, and deployable for in situ and real-time measurements. Okay, let's start from the very basic approach of the spectroscopic technique, the tunable diode laser absorption spectroscopy. So basically, you have a gas sample and you shine your laser through the gas sample. You have a transmitted light, which is ruled by the Lambert-Beer law. And you have that your input intensity is multiplied by an exponential depending on the absorption coefficient, which is depending of course on the oscillator strength of the transition that you're exciting, and the interaction path length, as you can see from the Lambert-Beer law here reported. This is the very basic approach, but since we want to be sensitive to very small variation of the incident light, And so we have to analyze to deal with the residual light beam with better increase the interaction path length. So what we can do from the single pass, we can go to some kind of solution involving mirrors in which you have a pattern of multi-reflections, as you can see here. Okay, and by employing this kind of configuration, you can use and you can implement Harriot cell or white cell, in general, multipass cell. Remember that here you have to have a light detector. A light detector in the different wavelength range from near infrared up to terahertz. So basically we are talking about photovoltaic, pyroelectric detectors, as well as bolometers, more fancy ones. And the cost, remember please that the cost of these detectors are in this pretty much in the same order of magnitude of the cost of the laser source. Okay, so if for the laser source you have 10K, the average cost of the light detector can go from 2 to up to 4K. But if you use very fancy bolometers, you can go much higher than that. And here you can see a very, very exotic configuration. This is a sensor that we built in when I was working in Rice University during my PhD. I spent one year and a half in Rice University, and this is one of the experiments that we did there. And here we employed a multipass cell and interband cascade laser. Sorry, is here. And of course a detector. This sensor was devoted to methane and ethane detection, and we were able to go down to 5 parts per billion and 8 parts per billion for methane and ethane respectively. Okay, so in this configuration we focus our attention on increasing the interaction path length. Okay, there is also other kind of approaches, just like the photoacoustic spectroscopy. The photoacoustic spectroscopy was discovered by Alexander Graham Bell in the 1840. And the basic concept behind the photoacoustic spectroscopy is the absorption of the laser light, of course. But if you are able to modulate the intensity or the wavelength or of the laser light, what you do, you can create a sequence of absorption of the light and then relaxation of the energy by the molecules. Okay, let's assume a train of pulses, for example, of the laser light. This creates an absorption and relaxation energy by the molecules. And this means that you are heating up and then you let cool down these molecules. So it means that the density of the gas is going to change. And if the density changes, you are creating a perturbation throughout the volume in which you have your gas, and basically you are creating a sound wave. Okay, and if you use a resonant cell, resonant with the wavelength of the sound produced, and you use microphone in order to hear the sound produced by molecules, you can relate the photoacoustic signal to the concentration of the gas target. Okay. Here is represented an intensity modulation or a train of pulses. Here we have an alternative approach for creating a modulated absorption of the light. This is called wavelength modulation. So basically, you don't modulate the intensity, but you rather modulate the wavelength emission of your laser. And thus, it means that you are modulating the absorption through across the absorption feature that you are exploiting. This is an absorption feature related to water vapor. Okay, and the photoacoustic signal in this case is proportional to the oscillator strength of the transistor that you are considering, the concentration of the gas, the power that you are employing, and the radiation to sound conversion efficiency. What does it mean, this last parameter? It means that when you have a process of energy accumulation and then relaxation, you need to be sure that the modulation frequency is low enough to allow the molecules to relax all the energy. So if the level of your target molecules compared to the level of the molecules in the gas matrix allow you to have a very fast a very fast energy relaxation, it means that you can afford also higher modulation frequency that in principle helps you in lowering down the noise. But if your relaxation rates for some kind of reason related with the energy levels are pretty low, you need to consider the possibility to lower down your modulation frequency. Otherwise, you will not give enough time to the molecules to relax all the energy. And this means that your process of heat generation, of your sound generation, is not effective. Okay, so here I'm going to present you a further evolution of this technique. And here they come, the tuning forks. So the tuning forks are those tools that you can use for tuning your music instrument, you know? And but here are used in order to sense the sound produced. So instead of the microphones that have a very, very large detection bandwidth, here you have a tuning fork. And why a tuning fork? Which is the improvement? The improvement is that you can rely on a very, very narrow bandwidth. Okay, now we will see. So basically, what does it change in the experimental configuration? You have your light sources, you focus your light sources, your light beam between the prongs of the QTF, and you modulate your light, just like in standard photoacoustics, okay? You create the sound, and you modulate the light at the resonance frequency of your tuning fork, okay? So this replaces the role of the resonant cell, because the resonant element is the tuning fork itself, okay? And the mechanical vibration that you are creating by, let the sound propagate and hit the internal surface of the QTF prong, you are creating deformations. And if you use a piezoelectric material, just like quartz, what are you doing? You are creating deformation, and you are creating also an accumulation of charges, because this is the most important characteristics of the piezoelectric materials, okay? And the first employment of the standard QTF in laser spectroscopy, laser gas spectroscopy, was made at Rice University in 2002 by Anatoly Kosterev and Frank Tittel. The first QTF they used was 32 kilohertz standard QTF, which is normally employed in all the watches and smartphones. Okay, for timekeeping, of course. Okay, here you have a schematization of the piezoelectric effect. This is the direct one, and here the converse one. What does it mean? That if you create a deformation of the QTF prong by putting it in motion, you will create an accumulation of charge. So it means a polarization, okay? And by applying some gold contacts here, you can collect these charges, and you can have a piezoelectric signal, okay? You can also implement a converse piezoelectric effect. So basically, you provide a sinusoidal signal, and thus you can induce the mechanical vibration, okay? It works in one way and the opposite way as well, okay? This is the fundamental vibration of a QTF here. Here we can see that we can describe the QTF in two ways, with electronics modeling or with a mechanical modeling. It's just the same, because you can model the QTF just like a damped oscillator under a viscous friction. But in the same way, you can represent the QTF just like an RLC circuit, okay? And as you can see here, the motion and here the law that put you in relation the voltage with the electrical characteristics and the charge created on a QTF, they are pretty similar, okay? Here you can find the damping parameter, and you can put in relation all the elements from both equations. So the motional capacitance here represents the mechanical elasticity. The motional inductance is the mechanical inertia. The motional resistance are the mechanical losses, and the parasitic capacitance that you don't want it, okay? But of course, when you are going to deposit electrodes, holders, and leads, you can get some of these parasitic capacitance, okay? And if you solve this equation, you have what? A resonance frequency. And this resonance frequency is related to the inductance and the capacitance, okay? And the resistance as well. If we talk about the quality factor, okay? So the frequency depends on the inductance and capacitance, while the quality factor on inductance, capacitance, and resistance, okay? This is the resonance curve of a 32 kilohertz tuning fork. In particular, this is the resonance curve of a tuning fork in damped conditions, because here we have a pressure of 50 torr, so it's not vacuum, okay? And the quality factor from the physical point of view is the inverse of the losses per cycle, okay? And you can calculate it by dividing the frequency by the full width of maximum of this curve. And the quality factor is also a good parameter to evaluate the selectivity in terms of external noise of your tuning fork. So it means that the tuning fork is sensitive to frequencies in this range, okay, from 32.762 kilohertz up to 32.6766, okay? This is the full width of maximum, 2 hertz. For what concerns all the external sounds of this full width of maximum, the QTF will not hear anything, okay? So let's go to see the merits and main characteristics. Of course, if you employ QTF as a sensitive element, you can realize very, very compact sensing modules. These sensing modules are called acoustic detection modules of few cubic centimeters. As I just said, the dissipative losses are extremely low. And of course, this is very, very important. We avoided the need to employ an optical detector, okay? Because the tuning fork is the sensitive element. You don't need an optical detector. You can rely on a very wide dynamic range from part per trillion up to percents. And of course, you are very immune to environmental acoustic noise. But there are some drawbacks which are common with other spectroscopic technique. The sensitivity scales with the laser power, and of course you have cross-sensitivity issues. It means that when you are going to detect given molecules, you need to evaluate all the possible interference species with these molecules. Of course, you have some kind of alignment cost, and responsivity depends on the molecular energy transfer process, as I just said. And we have been able to reach a record sensitivity in detection of 50 parts per trillion for sulfur hexafluoride. Okay. And the next step, starting from 2002 and coming to our latest research, was the design and realization of a first generation of custom tuning forks. So basically, we started from the design of the 32 kilohertz tuning fork, and we start designing and producing our own tuning forks. Okay, and here we have an example of a few of them. This is a 14 kilohertz, a 7.2 kilohertz, 8.5, 3.4, 2.8, 4.2. Okay, and also the dimensions are a little bit different. We have two crystal thickness. In particular, we have 0.34 micron, actually three, 0.34 micron, 0.25 up to 0.8, which is QTF number 6 here, quartz tuning forks. And we start from a Z-cut quartz wafer. We do etching by standard photolithographic techniques, and we also deposit a chromium layer in order to help the gold layers to attach to the surface of the tuning fork. And we have a very, very wide right now. Just like I told you, this is a very small set of tuning forks. Right now we have up to 20 different frequency that we can employ in photoacoustic experiments. Okay, let's go back to fundamental laws for what concerns the free motion conditions. The free motion conditions are described by the Eulero-Bernoulli equation here, in which we have the Young modulus, E, we have the moment of inertia, we have the section of the prong, and we have the quartz density. So if you impose the clamped free boundary condition, which means that you have to clamp one of the hand, and you have to let the other hand vibrate freely. By imposing this boundary condition, you can get the eigenfrequencies, and the orders of these eigenfrequencies turned out to be odd numbers, odd integer numbers. Okay, 1, 2, 1, 3, 5, 7. This is the fundamental flexural in-plane mode. Okay, in-plane means that the QTF progs are vibrating in the QTF plane. And here we have the vibrational profile. As you can see, we have an antinode here at the QTF top. The Kuiper signal is proportional to the quality factor of your resonator, to the absorption coefficient. In particular, the absorption coefficient takes also into account the concentration of your gas target. So actually is the oscillator strength here, S, the power of your laser, and as I told you, the radiation to sound conversion efficiency. And then we go to the first overtone mode, which is obtained by putting here the number 3 in the order n of the eigenfrequency. And as you can see here, you have two antinodes of vibration, one at top and the other one in the middle. And for QTF2, 4, and 5, which means the 7.2, the 3.4, and the 2.8, we got these overtone frequencies. So it means that you can use the QTF at the fundamental vibrational mode, but also at the overtones, depending on your application and on the modulation frequency that you want to implement. Okay? And here is just a confirmation that if you move your laser sources from the top of the prong to the base, and you are exciting with your cylindrical sound wave different vertical position, you retrieve this curve of the Quepas signal, okay, which is pretty similar to the simulation of the motion of the prong. Why the maximum is here instead at top of the QTF? Because actually, when you have your laser here at the top of the QTF, at least the half of the sound wave produced is lost here. And you can see this effect here in the signal retrieved with respect to the simulation that doesn't take into account this effect, okay? And then you can ask me, so you have been able to reach 50 ppt of sulfur hexafluoride as detection limit just with a bare QTF? No. Actually, we used a trick. We use an organ pipe, just like an organ pipe. If you use an organ pipe and you create sound within the organ pipe, you can create standing waves. And the wavelength of the lowest frequency of the standing wave here in the organ pipe is twice the length of the organ pipe of the 1D resonator. So the trick is like that. We can create a tube with small slits here in the middle, and we can put these tubes, we can align this tube between the prongs of the QTF in order to create a point sources of sound. And we can take advantage of the antinode given by the pressure wave here in the middle. As you can see, if you cut your organ pipe, your 1D resonator, for creating the slits in the middle, the deformation of the pressure wave is just very, very little, okay? And we got an enhancement thanks to this trick of the signal, of the photoacoustic signal of 130. And the overall length of this tube for a 7.2 kilohertz QTF is almost 25 millimeters, okay? Here you can see the quartz-enhanced photoacoustic spectroscopy compared with the standard photoacoustic and the TD-LAS sensor. So basically those sensors relying on multipass cells, and the cavity-enhanced base sensor. Those kind of sensors in which you create a standing wave of the electric fields, okay, in order to increase the build-up power within your cavity. So in this case, you increase the interaction packline. In this case, you increase the power. And we use in this graph a parameter which is called normalized noise equivalent absorption, which is a parameter that allows you to compare all the techniques by normalizing the performances with respect to the oscillator strength, with the optical power employed, and with the integration time. Okay, and as you can see, so you can understand that the lower you have of this parameter, the better are the performances. And here you can see that the quartz-enhanced photoacoustic spectroscopy is able to reach at 10 to the minus 11 normalized noise equivalent absorption, and with a very, very small effective optical path length. So it means that in order to reach comparable performances, these techniques has to employ a higher interaction path length, which means that increasing the size of your sensors, okay? And if we talk about the performances of the quartz-enhanced photoacoustic techniques, here are summarized the best detection limits. So we have 10 parts per trillion for HF. We have the 50 parts per trillion of sulfur hexafluoride. Here we have all the carbon compounds, the nitrogen compounds, hydrocarbons, alkanes, and stuff, from the near infrared up to terahertz. So if you don't believe me that the quartz-enhanced photoacoustic spectroscopy actually works, I will give you a very, very brief overview of the most important milestones in CAPA's research. In 2012, we demonstrated the sulfur hexafluoride sensitivity record of 50 ppt. And then we realized the first nitric oxide breath sensors. So this is a QEPA sensor, and this sensor was embedded in these bigger structures, in this bigger apparatus for sampling of the human breath. This guy, this wise old man, is Robert Kerr, the Nobel Prize for Chemistry. And here we have the nitric oxide concentration. This is a reference level of 100 ppb, and here we have the dynamic of the breath. In 2013, we had the demonstration of the first QEPA sensor in the terahertz range. The terahertz is a very convenient spectral region because the spectral feature of the molecules are pretty separated, and the rotational levels show very, very fast relaxation time. So the creation of sound is very effective in this range, and this discovery was big enough to deserve the cover of APL. And then we demonstrated the intracavity QEPAs. So basically, we used a bow tie cavity to build up the power of the laser, and we coupled with this cavity a quartz tuning fork. And we have been able to improve the single pass QEPAs of 240, more or less. And then in 2014, we demonstrated the first leak detector based on sulfur hexafluoride for mechatronic components. And then we demonstrated the first environmental monitoring sensor for CH4 N2O detection in Houston. In 2017, I spent 3 months in Aramco service company for a special project. In particular, I developed methane, ethane, and propane sensors, and also methane isotope sensors. And here, this shoebox that we realized is here embedded in a rack in which we have also a super sophisticated sampling system. And in 2020, we realized the first sensor employing an array of 32 quantum cascade lasers operated in pulse mode for broadband detection of CH4 and N2O as well. Here you can see the emission of the 32 lasers, okay, which were, grown from this substrate, and the spacing from each laser was 100 micron. And here you can see the reconstruction of the P and R branches of the N2O, and here you can see how the presence of methane can modify the overall spectrum of N2O. And in 2021, thanks to the European project Passepartout that we won last year, we are working on the implementation of QEPA sensor on drones in order to teach the drones where to fly. So drive the drones when the QEPA sensor is able to find some gradient in concentration of, I don't know, CO or methane or ethane. So basically drive the drones where the source of the leak is, okay? And then we will implement a network of drones. And let me tell you the last thing. There is also an alternative use of the QTF in laser spectroscopy. Rather than using the QTF as a tool to hear the sound of molecules, we can use the QTF to replace the photoacoustic, the light detector in the TDLS sensor. Why? Because we can directly focus the QTF, the light beam on the QTF, and we can create, through the residual beam interaction with the QTF, a photothermal energy generation. So basically, we focus the beam at the base of one of the prong, and we create a modulated heating and cooling of the quartz structure itself. So we create a process of energy conversion from the laser to the thermo energy to the elastic energy. So we induce the vibration and we create again the piezoelectric charge. Okay, this is an alternative implementation of the QTF. Okay, here there is the detail where we focus the laser beam. Actually, here there is the maximum of the stress field for the QTF motion. Okay, and here, and if you focus the laser light here, you can create the maximum strain field. Okay, this application is very important. We are investing a lot in this application because with respect to CAPAS, you are totally independent from the response of the gas matrix. And this is a non-contact technique because the QTF is totally separated from your gas. So you can use also aggressive and corrosive gases. It doesn't matter because the sensitive element is not immersed into the gas, okay? And of course, you can potentially replace the commercially available detectors. This is huge, and we are working hard on that. Let me spend 2 more slides about the Polysense Lab. The research activity that we have are totally devoted to gas spectroscopy. So breath analysis, environmental monitoring, leak detection, hydrocarbon gases monitoring, and monitoring of hotspot areas. We do sensing on demands and third-party consultants. If you are interested in contacting us, please visit the website polysense.poliba.it or my personal webpage here. And this is as group. This is our group of research. As you can see, we have a lot of PhD students, also thanks to the Optify project, which is a Marie Curie project network that we won last year. So thanks for your attention. And if you have any question, please do not hesitate.
Okay, perfect. Thank you very much, Angelo, for an excellent talk on some really interesting work. If anyone has any questions, please feel free to either raise your hand or type them in the chat. And we do have one question from Utkarsh, who says, thank you for such an insightful presentation, Angelo. I was wondering, is there a correlation between the Q factor and the modulating frequency?
Yes. Actually, we demonstrated, I mean, they are not directly correlated, but since the modulation frequency of the QTF, the resonance frequency of the QTF is proportional to the thickness of the prong, and is inversely proportional to the square of the length of the prong, and the quality factor is also depending on the thickness of the crystal, the thickness of the QTF prong, and inversely proportional to the length of the prong. Since you have this dependence on the dimensions and sides of the QTF, somehow these two figures of merits are correlated. In particular, I can tell you that if you increase the frequency, the resonance frequency of the tuning fork, you can increase also the quality factor. So the higher is the frequency, and the higher is expected the quality factor as well. Actually, the evaluation of the quality factor starting from the geometrical characteristics of the QTF is not easy, and also the simulation in COMSOL are not easy. So it's more like a phenomenological study, okay?
Great. Utkarsh also asks, could you please tell him how powerful the lasers that were used are?
Oh, yes. So we use, actually, we use very unconventional terahertz sources. And so we deal in that case we deal with microwatt, but in that case the oscillator strength of the features that we target are very, very high, up to 10 to the minus 17 centimeters per mole. But if we go in the mid-infrared range and you use quantum cascade laser, you can go up to also 1 watt of optical power, but the oscillator strength of the absorption are a little bit lower. okay? And if you go in the near infrared, we are talking about tens of milliwatt.
Okay. Also, just very quickly, I was curious. Have you explored the lifetime of these QTFs before? Like, are they?
Oh, this is a very interesting
there,
question.
or, yeah.
This is a very interesting question. So the aging of the QTF have been studied by people that works in timekeeping application. And, the damages, the aging of the QTF is in the order of magnitude of tens of years. But if you use QTFs in gas spectroscopy and you expose the QTF to toxic gases, to aggressive gases, just like H2S, you need to be ready to swap your QTF, because if you use high concentration of H2S, for example, just like 500 ppm, you will corrode your electrodes, and after that the quartz as well. So it depends on the application. Okay, I lost you for a second. Okay. Oh,
sorry, can you hear me now?
Oh yes.
Sorry,
yes.
I think I disconnected briefly there. Okay, great. Thank you for answering those questions, and thank you very much for your presentation once again.
Okay, thank you so much.
Thank you. So just before I introduce our next speaker, I'd like to let you know that after the next talk, there will be a brief quiz on all things optics related. There is a prize for the person who scores best in the quiz, which is a £50 hamper of Scottish produce and goodies, which I'm told includes a live haggis as well. If you are interested in taking part in the quiz, you need to go to this website that I've posted in the chat, kahoot.it, and input that PIN, and that should take you to a browser where you can join the quiz. So I'll put that up now and let people slowly trickle in, and then that quiz will take place after our next talk. So our next speaker is Dr. Jano van Hemert, who directs research at Optos, an organization devoted to developing novel technology for eye diagnostics and healthcare. He received his PhD in 2002 from the University of Leiden in the Netherlands, and he was admitted into the Young Academy of Scotland in 2011. His talk today is titled Imaging of the Human Eye. So whenever you're ready, Jano.
Okay, thanks very much. Is this visible? That's always the first question.
Yep, yeah, we can see that, and it's full screen.
Yep, can you see full screen? Good. Okay, hello everyone, and thanks for attending my presentation on imaging of the human eye. I'd like to have a bit of interaction in my presentation, so can you please type yes in the chat window if you knew about Optos before this seminar, and type no if you did not. I just have to briefly pop out to see if it's coming in. There's quite a few yeses already. I'm surprised. Of course, Andy, yeah. Everyone, okay, very good. A lot of people don't know that Optos is a part of Nikon. Most people know Nikon from its high-end consumer cameras. But they also have several other divisions, and one of these is the healthcare division that started in 2015 when they purchased Optos. Before that, Optos was a public company on the London Stock Exchange. It was founded 30 years ago in Dunfermline in Scotland. In this presentation, I will briefly introduce our main product and then show examples where Optos undertakes research with academics and doctors. So who is Optos? Optos develops, manufactures and sells medical devices for eye healthcare in over 55 countries. You can see the map underneath the text there. We now have more than 19,000 ophthalmoscopes installed globally. And we have 250 people out of 500 working near Edinburgh, which is our main headquarters. And in our headquarters, we've got all our R&D staff, 76 of those in product development. They will always work on the next product or the next product iteration. And then 14 in research, which is my team, and they pioneer technology for eye healthcare. We are well known for in eye healthcare for being the only company that produces ultra wide field images. A definition of ultra-wide field is in the article cited in the bottom left. The image in the center is a 220-degree optos montage of a healthy retina, and that equates to capturing 95% of the retina. The image to the right is a typical fundus camera, and that captures around 15% of the retina. And you can see the striking difference in how much of the retina you can see with our cameras. Over the years, we've diversified our products, but from an optical point of view, they all come from the same source, so to speak. Here I've placed them along two axes. If you go from left to right, we go from products being used in optometry to products used in ophthalmology. And the difference is that in ophthalmology, invasive diagnostic methods are included. That's where a fluorescent dye is injected in the arm of a patient, which flows through the retina and can be imaged by exciting it with a laser. At the bottom, we have added optical coherence tomography, which is used to create three-dimensional views of the retina. It is often abbreviated to OCT, very unhelpful, as searching for it generally gives you things that have happened in October. Here, all the imaging modalities supported by our flagship product, the Silverstone. The top row is reflectance imaging. That's a combination in our products of two lasers swept across the retina in less than half a second, capturing more than 12 million pixels. Red 3 is the green laser, and choroidal is the red laser. As red is a longer wavelength, it penetrates deeper than green. The reason it's called red 3 is because normally in fundus cameras, people use white light and they use a filter to obtain basically the retina by eliminating the red. The middle row are fluorescent modalities. AF is autofluorescence of the tissue with a green laser. FA and ICG are injected dyes with a blue and a near IR 800 nanometers respectively. And there you can see in the middle, the Optomat FA, how, for example, in that patient, the dye is not flowing to the left of the image, only to the right. And that patient has a very severe lack of perfusion of the retina because the blood and the oxygen is not able to get to that part of the retina. The bottom is a wide field optical coherence tomography image, and we'll get back to that later. Just as important as our medical device, just as important as our laser products is the medical device software called Optos Advanced. This software is called a PACS, a picture archive and communication system. It runs as a web server and enables the practitioner to use their web browser on any platform to log in to see their patients. Using an international standard called DICOM, Digital Imaging and Communications in Medicine, this software can handle any ophthalmic data from all compliant vendors. Here you see a typical screenshot with our ultra-wide field of reflectance image. Remember, that's the red and the green laser basically just reflecting in the top left with some green annotation on it. In the top right is the autofluorescence image showing the presence of natural fluorophores in the eye, and these can be overlaid if necessary. In the bottom left is an OCT B-scan image. B-scan because it is made up of a sweep of a laser where each column is referred to as an A-scan. If you start combining B-scans into a volume, people will refer to it as a C-scan, a terminology that we've inherited from ultrasound. The OCT scan here was created using our Monaco product, which uses spectral domain OCT. Our Silverstone product uses swept source OCT, which is more recent and for now also more expensive. Finally, in the bottom right is an image that ties the top and the bottom together. The green line shows where the OCT scan that was on the bottom left was taken on the fundus image. So that all seems to work, job done. Why then would Optos employ a team that has the pleasure to focus on research? The field of medical devices is the most competitive area of innovation. It attracts the most patent applications and has a constant flow of upgraded products. This innovation is driven by a healthcare system that's under pressure to deal with more and more patients and by a need to understand better the diseases and treatment. Optos, therefore, has to bring new products and features to its products to remain competitive. These features start with a challenge or an opportunity that bring much uncertainty about the efficacy. Does the technology answer the question? Is it accurate? Is it precise? Is it sensitive? Is it specific? Here, the research team starts by creating prototypes. followed by patent applications to protect that innovation. We then assess the prototype, most often in a clinical setting. The results are often submitted to scientific journals, and if sufficient positive evidence exists, we then start a handover into product with the aim to ensure the product performs as the prototypes. And even after the first products are sold, we work with practitioners to evaluate the features in specific clinical context. And that was all very abstract, and the rest of the talk is simply examples of all these activities. Here's the first example. This project finished early 2020. It was funded by the European Union to 4 million euros and comprised a set of universities and companies in 4 countries. The aim was to build the first ophthalmoscope that can produce optical coherence tomography images with a resolution of 1 micrometer. To achieve this, the system must have a very broad spectrum. Here, NKT's tunable laser sources were used with bandwidth of 300 nanometers. Margaret, from my team, worked with postdocs and staff from these organizations to develop the components, build prototypes, and assist in the imaging trials. In the project, we promised to show results on an animal model, which you can see here. The image here shows clearly the vessels at the top, which leaves very long shadows underneath, a world's first. But we went beyond the project's promise by taking an image of the human eye. This is not as simple as replacing the rat with a human. Human eye imaging requires many safety features to be in place. Also, the human eye is at a different scale, and that requires optimization of the system. The resulting image shows clearly several separate layers in the retinal pigment epithelium, the brightest band visible in the image. In commercial OCT systems, these separate layers combine due to a lack in resolution. This allows practitioners to study diseases such as age-related macular degeneration, which originates in that layer of the retina and leads to distorted vision and eventually to blindness. The next example. This project involved Opta supporting a postdoc at the Physics and Astronomy Department of the University of Glasgow. The aim was to design and prototype a solution to improve the contrast of our images whilst maintaining a wide field of view, for which we are well known, and it's a unique selling point that we cannot give up. Due to the goal of the Opta's optical system to achieve a wide field of view, it is not able to reach a high level of confocality. The article shows how to introduce more confocal imaging by splitting the return path into confocal and non-confocal. Separate paths are captured by separate detectors, and the images of these are then combined to balance all contrast in the central region with not losing the peripheral region. As you can see in the images on the bottom right here, we're specifically looking for a better contrast around the optic nerve head, which is the bright circular region, and the fovea, which is the dark region to the left of the red square. Okay, time for some more interaction. I will teach you how to label a retinal image as a right eye or a left eye. I'm not going to explain how this works. I'm only showing you the examples here. So have a good look at the 6 images. I've labeled each image with the eye it was captured on. It's either a left or a right eye. So now that you've learned the difference between left and right eyes, let's see how good you are at recognizing the following images. Here's an eye. Predict which eye this was captured from, and then type your prediction in the chat window as either left or right. A few more maybe. Oh, very good. Quick look. It was a right eye. That was about 75% hit rate. Here's another eye. Predict which eye it was captured from. Again, type your prediction in the chat window, either left or right. Results coming in a bit quicker this time. People getting very confident. Let's have a look. Left, 100% correct. Of course, that was very easy because you thought it must be the opposite to the one we just had, and you were right. What about this one then? Last one. Type your prediction in the chat window. Is it the left or the right? Pretty good. Some uncertainty there. It was a left eye. Well done. Very tricky because this one has significant pathology that obscures the features you were most relying on. There's blood on top of the optic nerve head, and there's even some blood over the fovea. As you can see, it's not as easy as you may have wondered. And this is just a classification task with only 2 labels. We've optimized that. This project involved Peter Wakeford, a PhD student, co-supervised, co-funded with the Gravitational Waves Group at the University of Glasgow. Gravitational waves sounds unlikely as to be useful for retinal imaging, but the group are experts in finding signal in very noisy data. And medical image data can be very noisy for many reasons, such as what I've just showed you. Pathologies or poor image quality can make it very difficult to get the right information out of these. And this has resulted in published papers and patent applications. This even resulted in Peter obtaining his PhD recently, and he's now working in the research team. As you can see from the abstract on the screen, this method is able to have a really high sensitivity of 99.4%. And why is that important? Well, here's a picture of a show and a news bulletin, and this is the best result. It is now featured in every Optos ophthalmoscope that we sell, and this helps to improve patient throughput, as highlighted in the marketing news bulletin. Practitioners no longer have to add this information manually, which also prevents erroneous entries. Now, given that we have more than 19,000 devices installed, each capturing about 50 patients a day, that means this algorithm runs 2 million times every single day. And by saving 5 seconds per image, we save a total of 330 working days every day. Last example, here's an example of an email that reached me. As a sales manager, has asked product management for evidence that the claims of our new products are true. He needs it to sell our products to the largest eye hospital in the USA. They want to know if the visible retinal area is the same as our previous product, whilst the image sharpness has improved. Now, luckily for me, in a collaboration with Harvard, where we worked on diabetic retinopathy, we answered that question and published it in Nature Eye. So all I had to do was basically forward that published article to the sales manager, who can then relay it to the team at the Massachusetts Eye and Ear Hospital. And that meant the sale went ahead, which, because it's such a large hospital, means several products installed. That's always a very good day for me. Just to summarize, so why did I give you these particular 4 examples? They all have a particular attribute in terms of the scale. Obviously, the Galahad, the first project, the Horizon, is very large, many partners, lots of postdocs and staff involved. But then also, for example, the PhD student, Peter Wakeford, 1 person involved, funded by the UK, much smaller scale, yet can have a really large impact, as you saw, as soon as the results of such a PhD studentship start hitting the product line. But also sometimes without any particular governmental funding, we do projects directly with teams such as I showed with Harvard. Also, I tried to get a range of projects that really covered that slide that I showed, going from all the way from uncertainty to getting things into product, and even doing research with features that are already in product. So after 9 years of a research team, what's the status quo? What are our results? I think the most important are the product features and the improvements, which we've done 31 so far. These drive more revenue into the business. But we also need to have a large patent portfolio to protect our intellectual property. As I explained, this is a highly competitive sector. And to date, we've had 288 patents granted. Our team members, they are often recruited from university, as they progress their career, they often do so by taking up positions in the product development side of R&D. And so far, we've had 8 of our research staff move on to product development. And the best way of showing the performance of our products is by applying the scientific method and getting the results of those methods peer-reviewed and then published in high-standing journals. And so far, we've had 45 of those. An important strategy to the team is to recruit talent, and we do that by developing that in conjunction with universities. As you can see here, 24 people have a degree where they work in conjunction, often co-supervised, co-funded by Optos. And that's really it for me. Here's a final slide with, if you have any questions, please do email me. We're also currently recruiting, and there's the link in the bottom right-hand side if you're interested in that. And if there are any questions right now, I'm very happy to take them.
Okay, thanks very much, Dr. van Hemert. That was a great presentation. Do we have any questions from the audience at the moment? You can either raise your hand or type in the chat. I was wondering myself just about, in the UK, what is the sort of uptake of this by the NHS? Is this a product that's mainly for private opticians, optometrists, or are you seeing it being taken into NHS hospitals as well?
It is taken into NHS hospitals. The original uptake was very low as Optos started out. And as a result of that, they started Optos Inc., which is the US version of Optos, and started selling into optometry, so more high street practitioners. And that's where the first success came from, and how Optos ended up being a public company. Soon after, though, we started focusing on the products that I showed more on the right-hand side, where you are looking at products that can only be used by practitioners inside hospitals. These are invasive procedures where you inject a dye into somebody's arm, and they come with a mortality rate. And these are the kind of products that go into hospitals, and that's when the NHS started becoming more interested. We also hope to soon have the products on the left-hand side, the more optometry products, to be approved for diabetic retinopathy screening, which is a very large UK program. In fact, the UK was the first one to start such a screening program. And as soon as we are part of that program, these will be rolled out also to screen diabetes patients annually. But yes, they are available in certain places in the NHS.
Okay, thank you. We've also got a question from Michele Lacerenza, who says, how long was the translation from prototype to fully implemented medical device?
Oh, it generally takes quite long. Optos was founded in 1991 as kind of a design company, and the first medical device was installed in 1999. So that was 8 years. I think we've improved on that a little bit, but it depends very much on your ambition of what kind of medical device you are building. Depending on what the device does, the kind of risk it has to the patient, it will be classified. And in the US, they have four classes: 1, 2A, 2B, and 3. 3 is generally very high risk. So when your life depends on it, a medical device would be classed 3, like a pacemaker, for example. A 1 would be something quite basic. Could even be a contact lens, something that does not exchange energy with the body. But as soon as you start exchanging energy with the body, then it becomes a 2A. So we are 2A device. And then you have to have a lot of documentation to prove that it's safe. And in recent years, also to prove that it actually performs as you say it performs. So you have some clinical trials and show with statistical tables that what your precision and accuracy is of the medical device. And that can add quite a significant amount of time to it. So think you've created your prototype. That may take 2 or 3 years. Then you have to do your clinical trials. That may take another year. And then you submit all your paperwork to the FDA, the Food and Drug Agency of the US. And then they have up to 9 months to decide whether you are allowed to sell the device into the US market. and they have to give you an answer within those 9 months. And that's only if you build a medical device for which you can already say a similar medical device is in the market. If that's not the case, if it's completely novel, you would have to do what's called a PMA, and that can take up to 3 years. So it can be quite painful.
Okay, thank you.
I don't want to put you off.
No, that's fine. Yeah, thank you again for the presentation. Also, thank you on a personal note, just because I'm a patient with type 1 diabetes, so I've actually used Optos devices in the past when I get my retinopathy screening. So
very good.
Thank you for the work you do, and thank you for the presentation, and thank you for those insightful answers to the questions. So we're going to have a quick break now for our quiz. I see 4 people have joined so far, so I encourage any more people to join using the following link, which you can open on your phone or in another browser, and then pin 749841. I'll just give a couple of minutes to allow people to join. Sorry, I've got the schedule slightly wrong. The quiz is not taking place until after our next speaker. I've just been informed, so apologies about that. So we will be having one more talk before the quiz, but I'll leave it open for anyone to join in the meantime. So again, apologies for that. Our next speaker is Dr. Fátima Domingues, who is a researcher at the Instituto de Telecomunicações at the Universidade de Aveiro in Portugal, which is also where she received her PhD in 2014 for her work on optical fiber properties and Bragg gating-based sensors. Her talk today is titled Optical Fiber Solutions for Physical Rehabilitation and e-Health Applications. So are you there, Dr. Domingues, and ready to start your presentation? Hello?
Hello, can you hear me?
Yep, I can hear you perfect.
I'm sorry, I was with a bit of troubles getting the sound and the video on.
No worries. Apologies again for getting the schedule slightly discombobulated.
No, that's okay. No worries, no worries. How can I share my screen? Okay.
So if you open the purple arrow in the bottom right and then click on the symbol, it's like a rectangle with an arrow coming out of it.
And should I? Which one should I share? The blank whiteboard.
If you go to share application / screen and then choose the PowerPoint application. Yep, there we go.
Okay, we are on. Okay, cool. So yeah, let me just pointer options. Again, sorry for the delay. First, thank you.
It's currently it's currently in presentation mode, so we can see the notes and things. Is it possible to put it full screen?
Yes, I just need to swap. Better like that?
Yeah,
perfect.
That's perfect. Right, thank you.
Okay, thank you so much. Sorry for the bit delay. And thank you for the invitation to participate in this event. It's always nice to be able to share our work. I'm from Instituto de Telecomunicações in Portugal, and I will give you a small overview, not a small, but kind of overview of the work we have been developing regarding optical fiber solutions for rehabilitation and the health applications. This presentation, I will give you a small introduction of all the work done. Then I'll focus on 2 kind of optical fiber based sensors that we are using and applying for our targeted applications. They are fiber gratings and Fabry-Perot interferometers. The first, you heard a bit about it in the previous talk from Dr. Rogério Nogueira. I will focus a bit on the production of the Fabry-Perot sensors, and then it's their application towards the complete gait analysis of human beings. Then I'll make a brief introduction to both of them, and then I will conclude and present some challenges within this kind of monitoring. So our work started, giving you a time lapse of what our work has been being developed. We started with the production of an insole containing several fiber Bragg rating sensors to monitor the plantar pressure. And after that, we thought, why not monitor all the gait analysis? First, I should, maybe some of you doesn't know what gait analysis stands for, because it's some kind of more technical term. But gait analysis is basically the way we walk. It's the analysis of the pattern that we have when we walk with a certain cadency, with a certain speed, and it involves not only the plantar pressure that we put on the sole of our foot, but also the angles involved in the ankle and at the knee level. So to monitor all these parameters, we developed several devices, and which I will go in a bit detail a bit further. So what kind of sensing mechanisms or sensing units did we use? For some we use, or for the starters, we start this work using fiber Bragg ratings. As said before, fiber Bragg ratings, they are structures that are fabricated by inducing a periodic change in the refractive index of the optical fiber. And we evolved from these kind of sensors to use a different kind of interferometical optical fiber sensor based on Fabry-Perot microcavities that are produced within the core of the optical fiber. These kind of sensors can be produced in several ways. It is very common to be produced through micromachining of the optical fiber, either using phantom second lasers or even using the splicing between different optical fibers. In this situation, for this one, we develop a method that consists in the recycle of an optical fiber that is previously damaged by the fiber fuse effect. The fiber fuse effect is a phenomenon that occurs when optical fibers are subjected to high powers, high density optical powers, and it encounters and it occurs in the presence of small bands in the fiber, in dirties or contaminations in the end of the fiber. It occurs when there are points that can heat up the fiber till ignites its core. This phenomenon propagates towards the optical source, and it leaves in its propagation path these kind of voids along the optical fiber. These voids, they are periodical spaced, and they completely disable the optical fiber for optical transmission. If we use this fiber and we splice it to an optical fiber, a normal optical fiber, working optical fiber, we will create bigger voids in its core, which will allow us to produce microcavities that act like Fabry-Perot interferometers. So the principle of work of these sensors that we are producing. The fiber Bragg ratings, as also in the other talk we saw, the periodic change in the refractive index of the core will produce a structure that reflects all the optical signal that abides by the Bragg law and transmits all the other components. Within the optical fiber, when we change this periodicity of the grating, the wavelength of the Bragg wavelength will also shift accordingly. So when we distend, when we apply, let's suppose we apply a strain in the optical fiber, I will be increasing this periodic, this period between the gratings, and my Bragg wavelength will shift towards a red, a higher wavelength. On the opposite, if I decrease it, I will have the shift on the opposite side towards the blue side of the optical spectrum. With these capabilities of these kind of sensors, I can monitor any change in the optical fiber, any deformation on the optical fiber. Regarding the Fabry-Perot interferometers, the concept is it's not the same, but it's similar. It's based on the deformation that is applied on the optical fiber. We will also induce a spectral shift. Will induce a spectral shift of the FP transfer function. And by monitoring this spectral shift or components that are affected by this spectral shift, I can also infer what is happening in my sensor or in the parameter that I am monitoring. So with these two sensors, we use the fiber Bragg gratings to produce components, to produce specific instruments of analysis for the foot plantar pressure, for the ankle plantar dorsiflexion, and after for the knee angular displacement. These components, by being able to monitor all these three parameters simultaneously, I'm able to map the gait of a person, which is of particular interest if we are facing a rehabilitation scenario where I want to know, for example, what's the pressure that the person is putting on the forefoot or around the ankle, according to the evolution of its step, or what's the angle, the maximal angle that a person can do after surgery in the knee or in the ankle, and how is that evolving with time. So with fiber Bragg grating, we managed to put all these through all these three devices together and make a full map of the plantar of the gait. Regarding the Fabry-Perot, it's still a bit more in its infancy, let's say, but we were able to develop a device to monitor the ankle plantar dorsiflexion. So I will start to present to you all the components of the devices that we did with the FBG sensors. As I said in the beginning, we started, the first device that we developed was an insole to measure all the plantar pressure. This insole has 4 sensors that are embedded in an epoxy resin. We also developed the device. Actually, they are 4 different devices, 4 similar devices that can be displayed in different areas of the leg, namely around the ankle to monitor this ankle plantar dorsiflexion, around the knee, and also to be able to measure the quadriceps tension and relaxation along the walking. As we know, the FPG sensors, they are also sensitive to temperature, and therefore there is the need to include a temperature sensor to compensate these values in the measures, to make sure that the temperature is not influencing the results that we are retrieving, and if it is, to be able to adjust these values by removing the temperature component on each sensor. So going bit by bit on each one. For the foot plantar pressure, I should say that all these, sorry, let me go back. I have a total of 8 FPG sensors. They are all multiplexed in the same optical cable. Here you will see one by one. I will show you one by one how they were produced and their principles of analysis. But in the overall final device, we have one optical cable connecting all of these sensors. So for our insole, we used an insole based in cork. Why cork? First, because it's comfortable. Second, because it's thermal isolated, can act as a thermal isolator, and therefore we will not have to include the temperature compensation, and also because of its near Poisson ratio close to 0, which gives the feature that if I'm stepping on this point, for example, all the surrounding pressures will not be felt. This basically means that as I am walking, I will see each point activated in time and not all at the same time, and the pressure exercised in this area, for example, will not affect the pressure exercised in this area, or at least it will not be measured by this sensor. This isolation you can see in this graph here, when within the calibration process in which we were adding loads in one sensor in this FPG, you can see that there is a shift according to the loads applied, while all the other 4 FPGs that are in all the other points, they remain exactly the same. Regarding the monitoring of the monitoring of the plantar pressure with this device, we can see here in the graphic the first point that is activated. It's the FPG1 in this point, which is when the heel strike, when the movement of the foot. It's followed by all this at the foot flat movement. As we raise the heel and we put all the weight in the forefoot, these are the sensors that are getting activated. And at the toe off, you can see that the heel doesn't suffer any pressure anymore. It's already off, and the toe is now at its maximum peak of high. This cycle, it's exactly that. It's a cycle. This is just one step, and this is the layout. It's the results obtained for one step with one foot. But the gait, the walking, is the cycle movement, and we will have this all this pattern repeated sequentially. Any deviation of the typical pattern, that is a typical normal pressure distribution pattern, any deviation from it, we can deduce some pathologies. We can, regarding to the spinal cord, regarding to the foot anomalies, or namely ulcers, that can be developed, and will help us detect that this kind of device can help us detect that anomalies on an early stage and act accordingly to mitigate them. For the ankle plantar dorsiflexion and for the knee flexion and extension, we develop a system that is based on the kinesio tape attached to a sensor. First, the kinesio tape, I think you should know it or at least seen it with athletes, football or other kind of sports. That's the kind of tape that they use along the muscles to give muscle support. So it's a biocompatible and already in use tape for muscle evaluation and rehabilitation. What we did, we took our optical fiber with the recorded FBG. We embedded that FBG in epoxy resin block. This epoxy resin is liquid, so it's easy. We just have a mold. We insert the FBG in that mold, and we fill the mold with the resin. Is a thermosetting, as I said, is a thermosetting resin, and which means that when it dries, it gets in a kind of rubber state that can be flexible. We attached some hooks and adapters to this block of resin, and we adapted it. We glue it actually to the kinesio tape. This procedure allows us to reuse the sensor, but obviously the kinesio tape is at loss. It's not reusable, but the sensor can be unhooked and reused in another application. What it happens with the sensor when the kinesio tape is stretched? Also, the resin block is stretched inside it, which because the fiber is within the resin block, which will induce a wavelength shift to higher wavelengths. On the other hand, if it's compressed, the same will happen to the FBG wavelength. By monitoring this wavelength continuously, we can then monitor the movement or the compression or extension movements that are applied in this section of the kinesio tape. So application-wise, this kind of sensor, the kinesio tape, is glued along the ankle, this for the ankle plantar dorsiflexion. The sensor is located along the shank, and we have to make sure that the kinesio tape is long enough to cover the angular this angular area of the ankle. When the ankle is on a plantar flexion mode, the tape is stretched and I have positive wavelength shift. On the other hand, when it's on a dorsiflexion mode, the wavelength is compressed. The kinesio tape is compressed, which will compress also the sensor, and I will have a blue wavelength shift. With this device, we can therefore monitor the angular movement of the ankle. Similar to the ankle, we use exactly the same device, and we can also, if we put it along the extension area of the knee, when the knee is in a flexion extension mode, it will either tension or relax, compress the optical fiber, and by monitor also that Bragg wavelength, I can also monitor that movement of the knee. For the quadriceps, this is the first recap, the recap of the first image presented. The knee is the same. As we walk, we have moments of tension of the quadriceps and of relaxation of the quadriceps. When they are tensioned, the same will happen. The key tape will be on attention mode, and then it will induce the wavelength shift, the Bragg wavelength shift of the sensor. This is the results that we acquired for all the sensors deployed. We have here for starting with the hill. That is the first that it's activated according to the plantar pressure. It's following by the metatarsal, and the fourth metatarsal and the are almost simultaneously activated, but we can see clearly that the toe is the last one to be activated. Similarly, we can have it's more easy to compare the quadriceps activity with the movement of the knee, because they are actually correlated in between. And we can see that it is when the knee is an extension that the quadriceps are more active. And also we can see the movement of the ankle. The ankle plantar dorsiflexion is also very visible in the sensors. As regarding the temperature compensation, we did not perform temperature compensation in this data because the temperature variation was very low and doesn't affect the results. Just to highlight one thing. Regarding the plantar, the plantar force or the plantar pressure monitoring, it's more detailed to have it by areas, for example, by heel, fourth metatarsal, first metatarsal, and so on. But finally, we have a pattern to evaluate the pattern, the global pattern of that plantar distribution. Is generally what is done is all the forces in which area it's summed up, and then we will have a pattern similar to what is represented here. So this is what was made with the fiber Bragg grating sensors. As for the FPE sensors, just one quick remark. The FPG sensors, it's easy to multiplex it, because we have one wavelength or we have n wavelength Bragg wavelengths peaks that we can monitor simultaneously. Regarding the FPE sensors, we don't have one peak. We don't have one wavelength that is reflected. We have a spectrum, a spectrum of a certain order that we can analyze. This when we are working with a broadband signal, for example. So we needed to find a structure that allows us to take one point of one section of this spectral reflection from the FPE and analyze it on a continuous and not, we don't need a very high rate acquisition level, but we need acquisition rate that is higher than 10 samples per second, because one step is very quick. And we need to have enough information within that second to evaluate correctly all the pattern of the sensor. So to be able to use a lower cost kind of configuration, which is based first on a lower cost sensor, which is made with recurring to a splicing machine only. We chose to develop an interrogation system that is based on the convolution of the optical light source within the FPE interference pattern. By that, what did we do? We select a laser source, a narrow bandwidth laser source that we inject in the sensor, in the FPE sensor. And when we reanalyze the optical power that is reflected by the sensor by a photodiode, this optical power, which is this area, changes according to the wavelength shift. We can see that, for example, when the FP is in a relaxed mode, we have this area that is reflected is optical power. And when I apply a strain to the FPE, there will be increase in the optical power reflected. There are some constraints regarding this application, namely the area of detection, the linear area of detection, because I only have a certain, a given, let's say, a given area of detection that I cannot go all the spectrum. But it's definitely a low cost, lower, let's say, lower cost way of monitoring, and it gave us some promising results. We did the same. We used exactly the same device, and you can see here that when there is no flexion, I have this amount of optical power. And when there is when the ankle is in a dorsiflexion, there is an extension of the fiber, a tension applied to the FP, and the optical power is increased. We did some tests with these kinds of mechanisms, and as you can see, this is very easy to attach and to remove from the person that is performing the tests. And also we could acquire a very detailed signal of the plantar dorsiflexion during several steps that the volunteer performed. And also we can highlight that the resolution or the accuracy of the results are within the ones that we also obtained with the FPG sensor. So in conclusion, we present some optical fiber Bragg ratings that can be used to accurately monitor physical parameters related to the lower limbs movement. I also show you some Fabry-Perot interferometers that can be a cost-effective solution for the accurate monitor of the limbs and angle movements. And now which is the one that is, in my view, one of the highest challenges in the application of this kind of sensing units in e-health architectures. It's the cost of the analysis system and the development of systems that can be lower cost and as accurate as the ones in the market now, but which can make these kind of sensors a portable, not portable, but movable sensors that can give it the IoT, the e-health kind of label that they can they can do, that they can have with regards to the capabilities that they have in terms of accuracy, of detail that we can get from them. Thank you. That will be all.
Great. Thank you, Dr. Domingues. That was a wonderful presentation. I encourage anyone that has any questions to either raise your hand or type them out in the chat. And I'll kick things off with a question of my own.
Okay.
In terms of the data sets that this sensing system produces, the complete gait analyses, do they have to be analyzed by a human, or have you looked at sort of pattern recognition and machine learning and ways that algorithms can do the diagnoses?
At the moment, yes, but I'm sure there are already data sets that can be used to integrate with the data that we are retrieving. Because gait analysis is already a very wide field and very well studied. Yes, there will be for sure data sets that can be integrated and compared to the ones that are obtained with these kind of sensors, yes. Okay.
And also, I was curious, what are the current sort of techniques for assessing someone's gait? Like, do any sort of machines or sensors exist currently, or is it just a clinical?
Okay, you have the very, I will say, I will not say not accurate, but well, they are what they are. that are attached to your phone or to your watch, that you can study the cadency of the step, you can the speed and so on. But when you go to this kind of detail, that you want to know the pressure, for example, the plantar pressure in which person, or on the angle kind of level, or on the knee or on the ankle, they are more clinical for sure. You have sets that are based on video. There are for the plantar pressure, there are platforms that you can walk on. They are still very expensive, but they exist and they have some accuracy to it, of course.
Okay, thank you. Just one final quick question. So you mentioned the sort of typical step, and that a deviation is pathological. Is a deviation from that always pathological, or are there some variations in what's considered normal gait?
There is always deviations, and this means that, for example, the same person don't have the exactly same layout or same pattern that coincides 100%. There is deviations to it. There is deviations between one person and the other for sure, because of the weight, because of the way the distribution is made. For the assessment for the pathology or the evolution of the pathology, it's necessary to make a previous study, and the evolution study. For example, if you are making a rehabilitation exercise given to one problem that you have on the spine, so you will have the first assessment, then you will have all the rehabilitation process, including physiotherapy and so on, and you can target that evolution based on these devices. These devices can give you insight on is the rehabilitation going on the right way or not. Basically, that's the point.
Okay, perfect. Thank you very much, and thank you again for the presentation.
So hi everybody. I will moderate this session of the event, this last session. I hope everyone is hearing me. Drop a message on the chat if this is so. And so we will start. We will continue. So first of all, I will thanks all the organizers. We are talking about Cisma, who was taking the most of the organization side of the project, and OSA chapter of Milan, EPS Young Mind, FIJUA, Scope, and all the other organizer of this event. So it has been great. I really enjoyed all of the talks. And it was a long journey through optical sensing and new optical technologies, and it was great to see all the different applications, from material application to medical application. And I'm so excited to present you the next two speakers. So we will be inside a new part of this sensing world, mainly related to tissue optical acquisition, either talking about imaging on one side, and on the other one by local sensing. So the two speakers are Caterina Amendola from Politecnico di Milano and Calum Williams. The first one is Caterina Amendola, and so her science background starts many years ago here at Politecnico di Milano, and then he has passed some times, nearly half year, 1 year, working at the European synchrotron facility of Grenoble. For X-rays interaction with matter, also always directing to medical applications. Then he started her PhD in tissue optics, nano-optics, and photonics Photonics for Health at Politecnico di Milano, and she is finishing her PhD today. I mean, in this year. Her the final aim of her research is aiming at clinical application in intensive care units, both for oxygen saturation monitoring of tissue of patients in intensive care units, so adults and newborns. So the applications ranges from adult patients to newborns, and she will tell us something about the new application on COVID-19 patients. So Caterina, the floor is yours, and go on, inspire us.
Thank you very much, Michele. Can you hear me?
Yes.
I'm trying to sharing my screen. Okay. Okay. Can you see the presentation?
Yes, we can.
Okay. So thank you very much, Michele, and thanks to all people that organized this great event. I'm very happy to be here and share with you our latest results on the use of diffuse optics devices for monitoring tissue oxygenation and perfusion. So in the next slides, I will first introduce the importance of these two parameters, tissue oxygenation and perfusion, and the advantages that can be obtained by monitoring them through time domain years and diffuse correlation spectroscopy. So I will summarize you the physical principle of these two techniques, and then I will show you the hybrid device that we are developing here in Politecnico di Milano that combines 3D NIRS with diffuse correlation spectroscopy. In the end, I will show you some applications of these two techniques for monitoring the hemodynamic parameters of both neonates and adults. So why are we interested in monitoring tissue oxygenation and perfusion? Well, because the tissue oxygenation is the concentration of oxygen in the tissues, and oxygen is the fuel of our body. It's the fuel of our brain, of our muscles, of our tissue. So monitor the techniques that allow us to monitoring these concentration of oxygen is extremely important. On the other side, perfusion is the rate of blood that is delivered to the tissue. And these two parameters are strictly related by the fact that the oxygen is delivered to the tissue by the blood. In particular, red blood cells contains a protein that is called hemoglobin, which is responsible for the oxygen transport into our body. So when we breathe in, what happens is that a hemoglobin protein binds to an oxygen molecule. And we will call this particle oxygenated hemoglobin. Then red blood cells start to flow into our body, and at a certain point, the bond between hemoglobin and oxygen breaks, and the oxygen is delivered into the body, and we will call the hemoglobin protein that is not bind to any kind of oxygen molecule, the oxygenated hemoglobin. There are some pathologies that could impair this mechanism of transport of oxygen inside our tissue, such as acute respiratory distress syndrome, as the one that suffer COVID-19 patient, or anemia that is very common in preterm neonates. So in these cases, monitoring techniques that allow to measure the concentration of oxygen and tissue perfusion are extremely important. And nowadays, the most used techniques are positron emission tomography and functional magnetic resonance imaging, that however are bulky. They do not allow for continuous monitoring and require patients to stay still. That in some cases, as for example, when you deal with neonates, is an extremely challenging task. So in this scenario, near-infrared spectroscopy seems to be an advantageous alternative for continuous monitoring hemodynamic parameter at bedside. Near-infrared spectroscopy techniques are based on the concept that light can penetrate for a few centimeters inside our tissue because of the very low absorption coefficient of its main constituents, such as water, lipids, hemoglobin. So by studying the interaction of light in this region, so in near infrared region, we can retrieve information on the concentration of oxygenated and deoxygenated hemoglobin concentration. So it is possible because, as you see also from this image, their spectra is different in near infrared region. So in time domain years, we exploit laser pulses of 100 picoseconds of duration that are injected into our tissue, and we analyze the pulse that is re-emitted by the tissue, whose shape depends on the optical properties of the tissue investigated. So by fitting this re-emitted pulse with the diffusion approximation of the radiative transport equation, we can separately retrieve the absolute value of the reduced scattering and absorption coefficients at different wavelengths. And from the absolute value of the absorption coefficient at different wavelengths, we can measure the absolute concentration of oxygenated and deoxygenated hemoglobin. And from them, the total hemoglobin, that is the sum of the two, and the tissue saturation, that is the ratio between oxygenated hemoglobin and total hemoglobin. So, however, as you can see also from this image, if you are interested in measuring the hemoglobin concentration in the brain, light should pass through many different tissue types before reaching the tissue we are interested in, that are scalp, and cerebrospinal fluid. And here the advantage of using time domain years, because by exploiting an intrinsic information that is held in the re-emitted pulse, that is the arrival time of the photons, we can disentangle the information coming from superficial tissue and deep tissue. In particular, the early gate photons, that are the photons that arrive early, are the ones that travel less inside our tissue, and so they are more sensitive to the superficial part of the investigated media. Whereas photons that arrive late gate photons, they travel more inside our tissue, and so they are more sensitive to the deeper part of the investigated media. And so in that way, by exploiting these characteristics, we can disentangle the information coming from superficial from deep part of the medium. In diffuse correlation spectroscopy, instead, we use a continuous wave laser light, highly coherent in space, that is injected into our tissue. But the biological tissue, in biological tissue, there are many scattering centers. So what we will see in the detection plane is a speckled pattern that is due to the constructive and destructive interference of photons that are scattered by different scattering centers or that travel different paths inside our tissue. So in near infrared region, the scattered particles are red blood cells. So as you can imagine, they are not fixed in time, but they move inside our tissue. And so this will cause a fluctuation in this speckle pattern that we will observe. And if we fix on just one of these speckles, what we will see is that its intensity will change in time. We can quantify these fluctuations by computing the intensity autocorrelation function G2, which is directly related to the electric field autocorrelation function G1. And this last function can be expressed considering our tissue as a diffusive media and considering the motion of red blood cells in microvasculature as a Brownian motion. The electric field autocorrelation function can be expressed as a function of dB, the diffusion coefficient of the Brownian motion. So this quantity will give us information about the motion of red blood cell, and we will call it blood flow index. So what happened basically is that when blood flow is low, so we have a low blood flow, also this dB parameter will be slow, and so the electric field autocorrelation function will decay to 0 at high delay time, at high tau. But when blood flow increased, this dB parameter became bigger, and so the electric field autocorrelation function decay for lower delay time, for lower time. And so in that way, we can retrieve the information about the blood flow. But why combining these two techniques together? The reason is not only that in this way we can simultaneously measure tissue oxygenation and perfusion, but it is also that we can get an extra information that the two techniques separately are not able to give us. And this information is the tissue metabolic rate of oxygen extraction, which is the rate at which our tissue is extracting oxygen from our blood. As you can imagine, this quantity is strictly dependent on the blood flow that we can measure with the DCS and from the quantity of oxygen that our tissue extracts from the blood. And this second quantity can be computed as the difference between the concentration of oxygen of the blood that flow into our tissue, that is the arterial blood, and the concentration of oxygen that flow out from our tissue, that is the venous blood. And this second quantity can be measured by combining time domain years device with a commercially available pulse oximeter. So here in Politecnico di Milano, we developed a hybrid device that combines time domain years and diffuse correlation spectroscopy techniques and allow us for simultaneous measurements. It is a compact device, so it is held in a 40 by 40 by 60 centimeter cube, and it allows for measuring at one interfiber distance for the TD nearest modules and at two different interfiber distances for the DCS module. The TDS modules is made up of two pulsed diode laser emitting light at 670 nanometer and 830 nanometers. The intensity out from the laser is modulated by two variable optical attenuators, and the re-emitted pulse is collected onto a single photon detector that sent information to a time-correlated single counting board that allow us to reconstruct the re-emitted pulse, the shape of the re-emitted pulse. The device that we are using is a prototype of a now commercially available device, and it has been tested over 3 different protocols for TD near systems, obtaining very good results that are in accordance with state-of-the-art systems. In particular, it shows a very small variation of optical properties over 15 hours of measurements, lower than 1%, and variation over repeated measurements on 1 month, lower than 2%. On the other side, the diffuse correlation spectroscopy module is made up of a highly coherent continuous wave guide laser operating at 784 nanometers with a maximum power of about 100 milliwatt. The photons are then sent to an optical switch that alternatively directs the light into different branches. In the first branch, light is injected at a high distance from the detection position, and so we need a very high quantity, a very high power to be injected into our tissue. And to stay beyond safety requirements of maximum emittable power per unit area, we needed to split the signal into different injection points. On the other branch instead, light is injected at smaller interfiber distance, and so we just attenuate the beam with a variable optical attenuator. For the detection channel instead, we used a bundle of 4 single mode fibers that sent the re-emitted light to 4 single photon avalanche diodes. And these 4 channels are needed to increase the signal to noise ratio of the high interfiber distance measurements. The electrical signal of the detectors is then sent to an hardware correlator that allows us to reconstruct the intensity autocorrelation function. Also, the DCS module was tested over long measurements, so 3-hour measurements showing a variation of the dB coefficient lower than 5%, and over repeated measurements over 15 days with a variation lower than 3%. We also measured its ability in retrieving variation of the dB parameters and its depth sensitivity. Finally, we measured the crosstalk between the TD near signal and the DCS module, because in the TD nears module, an interferential filter is placed in the detection channel in order to cut off the light coming from the DCS laser. But in the DCS module, no filters have been used. And so we had to be sure that the signal coming from the TD nears lasers are not affecting our measurements. So what we did was to measure the DCS signal at different distances from the time domain near sources. And what we observed is that if this distance is higher than 16 millimeter, then the DCS signal is not affected by the TD near source. So in the probe that we are 3D printing here in Politecnico di Milano, we always use the distance between the DCS detectors and the time domain near injection higher than 16 millimeters. Now I would like to talk to you about two of the applications we are currently working on, where we are monitoring the hemodynamic parameter of both neonates and adults. So I would like to start to talk to you first about the application on neonates. But before going in details of our clinical application, we studied the accuracy of time domain nears in estimating the cerebral hemodynamic parameter of neonates. We did it using a mesh-based Monte Carlo tool to simulate photon propagation inside realistic meshes of neonate's head, as the one that is shown here. And in particular, we use two kinds of meshes, preterm, so 29 weeks post-menstrual age mesh of neonate's head, and 44 weeks PMA mesh. The head structures were divided in 3 different tissue types with different optical properties that are extracerebral tissue, comprehensive of scalp and skull, cerebrospinal fluid, and brain. The interfiber distance was set to 15 millimeter to reproduce the one used in our clinical measurements. And we also tried to simulate the intersubject variability of the optical properties by increasing and reducing the optical properties of the 25% in the different domains. Here I reported the results obtained analyzing the simulation with the semi-infinite homogeneous model for photon migration. That is the model that we use for our clinical measurements. And the gray line are the values that have been simulated in the brain for both total hemoglobin and brain saturation. And we tried to simulate an ischemic event that is characterized by a constant total hemoglobin and a reduction from 70 to 40% of tissue saturation. In red and blue instead, we can see the estimated value for respectively the term and preterm neonate's head. And as you can see, the brain total hemoglobin was underestimated. And this is probably due to the presence of the cerebrospinal fluid, which is a very low absorbing and scattering media. Whereas the brain saturation, oxygen saturation, was retrieved with acute accuracy and an error smaller than 10%. So being aware of that, we are currently using diffuse optics device for monitoring the hemodynamic parameters of preterm neonates that need red blood cell transfusions. Indeed, 90% of extremely low birth weight infants receive more than one red blood cell transfusion. And despite the advantages that can be given by this transfusion, many complications may occur. and it seems that the nowadays used biomarker, which is the concentration of hemoglobin in the blood, is not the perfect biomarker. So what we want to do in this clinical study is to measure the hemodynamic parameter, the cerebral hemodynamic parameter of these preterm neonates before and after the transfusion, to see the effect of the transfusion and try to find a new biomarker for red blood cell transfusion needed. So we use the BabyLux device, that is a device that has been developed for an European project with different partners, European partners. And this device combines time domain NIRS and diffuse correlation spectroscopy modules with just one interfiber distance for both modules at 1.5 centimeter. So for this small interfiber distance, just one detection channel is needed to obtain a good signal-to-noise ratio. In our protocol, we measured 5 repetitions of 60 seconds acquisition before and after red blood cell transfusion. What we expect from these measurements? Well, we expect that the red blood cell transfusion cause an increase of oxygen in the blood, and this should be observed by an increase of cerebral oxygenation and a reduction of the tissue oxygen extraction, which is the ratio between the oxygen extracted by the tissue from the blood with respect to the one that the blood can deliver. On the other side, we also expect a vasoconstriction that should be observed in a reduction of the blood flow index and a reduction of the metabolic rate of oxygen consumption. Here I reported the preliminary results of the first, let's say, the first measurement that have been done on 12 different subjects. And as you can see, the brain saturation and tissue oxygen extraction show statistically significant variation that are in accordance with our expectation, whereas blood flow index and CMRO didn't show any significant variation. And these results suggest us that the decreased cerebral oxygenation due to the anemic state is not so dramatic to affect the cerebral metabolism of these preterm neonates. However, we are working to enroll 18 more subjects to verify our hypothesis. The second application I want to talk to you about is an application where we are monitoring the hemodynamic parameter in adults. And in particular, I want to talk to you about the VASCOVID project, that is a European project that is started a few months ago, where different partners from all of Europe are working to develop a new compact, very small and wireless and also user-friendly time domain NIRS and diffuse correlation spectroscopy device for monitoring the microvasculature and endothelial health of patients that suffer from acute respiratory distress syndrome, such as COVID-19 patient. So how do we want to do this? Because in 2016 it has been observed that the hemodynamic response to occlusion protocol between healthy subjects and patients that suffer from acute respiratory distress syndromes is different, and that the response change with the development of this pathology. So in the VASCOVID project, we want to monitor the hemodynamic response of an occlusion protocol on the thenar muscle, which is the thumb muscle. But before doing this, we have to understand what is the hemodynamic response of healthy subjects. So Since the device that I showed you before is a prototype of what will be developed in the VAS-COVID project, we have done this at Politecnico di Milano. So we have measured the hemodynamic response of healthy subjects to the VAS-COVID occlusion protocol. And here you can see the results for one subject, where the gray part of the graph represents the occlusion period. And as you can see, during the occlusion, both the tissue saturation and the blood flow index dramatically decrease, going to 0. Whereas after the release of the occlusion, we can see a great increase in blood flow index, tissue saturation, and total hemoglobin. However, these results are just for one subject, and we are working for recruiting other healthy subjects to increase the statistical consistencies of these preliminary results. So in conclusion, I show you how hybrid time domain years and diffuse correlation spectroscopy devices can be used for monitoring the hemodynamics of both adults and neonates, and how these devices can be built to be small and user-friendly, to be suitable also for delicate environment as the one of the intensive care units. Finally, I would like to thank all people that collaborate with us in this study, from Politecnico di Milano, from doctors from Mangiagalli Hospital, and from ICFO Center in Barcelona. And I would thank you for your attention. Okay.
Thank you very much, Caterina. It was a great presentation, and it is amazing to see that photonics devices, optical devices, are actually used in clinics, in intensive care units. And so from research to translation and actual usage of those devices. I have a curiosity about using the devices in intensive care units. So you've got the experience, the live experience of it, of being in an intensive care unit while people are sick, or like newborns had to undertake red blood cell transfusion. And what should the features of those devices be in order to be useful in those kind of situations? And what are the pros of having those devices?
Okay. Thank you very much for this question. Well, working in this kind of environment is for sure extremely, I mean, from an emotional point of view, is extremely hard, because you are, I mean, you are in a place where every second there are monitors that sound, because some parameters of some patients are below some threshold, and so. From this point of view, it is extremely hard to work in this kind of environment. But on the other side, you can really see what are the needs of the doctors in terms of, for example, in the Babylux device, we have contact sensor that prevent light from to be emitted when the probe detach from the tissue, so detach from the head of the neonates. And this is an extremely important, it's something that if we didn't talk with the doctors, probably we didn't thought about using something like this, but it is extremely important, because neonates, I mean, you cannot say to neonates to stay still. He moves, he cries, they move, they cry, and so there are working in this environment, from my point of view, allow also to see all the things that can be modified to better follow the need of the doctors.
Yes. Okay. Yes. Thank you. Yes, you were talking also about like easy to use, that you're trying to move this device to an easy to use system in the VAS-COVID project.
Yes, absolutely. Yes, and this
Okay.
is the aim of this project, of the VAS COVID project.
Okay, thank you very much, Caterina. If there are other questions, please answer them on the chat.
Okay, thank you.
And we will then now move on to Calum Williams. And so his journey starts from Cardiff University, where he begins his studies. Then he moved to University of Cambridge to study micro and nanotechnologies, where he completed his PhD in plasmonics nanostructure for enhanced optical devices. Calum is now junior researcher fellow at Wolfson College at Cambridge, where he works to unify optical imaging modalities using nanophotonics. He also has research collaboration with major institutions such as University of Bath and NASA. And nowadays he's going to talk to us about the latest development of snapshot multispectral image sensor for biomedical imaging and its application. So thank you, Calum, for being here, and the floor is yours.
Thank you very much for the very kind introduction there. I think that's the kindest introduction I've ever received. So thank you very much. Let me just share my screen here, and thank you very much for having me. Right, let me just put my slides on. Can you see the? Let me just put it big. Can you see my slides?
Yes, perfect.
Wonderful, excellent. So again, thank you very much for the introduction, and thank you for the invitation and having me here. So yeah, so I'm a postdoc here in Cambridge in the Department of Physics. Let me just put on my little laser pointer. And yeah, this is my talk, kind of overview in the development of snapshot multispectral image sensors for biomedical imaging applications. So yeah, so let's kick off. So yeah, I'm a postdoc here in the group of Professor Sarah Bondique. The group is called the Vision Lab, and it's quite a diverse and multidisciplinary group. So I'm just going to kind of give a very brief overview or introduction here. And the general mission statement of the group is, to advance our understanding of tumor evolution using next generation imaging sciences. And that's done with kind of computer scientists, with physicists, with chemists, etc. And we're split between here in the Department of Physics and down at the Cancer Research UK Cambridge Institute. And there are a lot of research strands in the group, everything from optoacoustics through to endoscopy, multispectral filter arrays or spectral imaging in general, through to kind of machine learning techniques, kind of trying to model different biophysical phenomena or interpret different results, all the way through to kind of preclinical studies in different cancer models, and also kind of first in human clinical trials. So there's a range of different aspects here. My kind of expertise, if you will, or research interest is in this area here. So I'm primarily going to only really talk about this area here. So I guess just to kick off, I mean, so what's the problem and why should you even kind of care about what I'm going to talk about? And the general area that I'm going to talk about and describe the problem is endoscopy, and in particular, upper GI endoscopy. So I'm sure a range of people are familiar with this, but just to kind of overview, the general statement that cancer survival rates are typically higher if that cancer is diagnosed early or early enough. But doing so, this diagnosis, that's the difficult part. And there are a whole multitude of different Cancer Research UK statistics that are around, but typically, so the 10-year survival rate for 8 different cancers combined here, it's much greater if it's diagnosed at, say, stage 1 or stage 2. So your survival rate is much higher, 3 times higher when it's diagnosed early compared to where it's diagnosed late. So why can't we just do that? Why can't we just diagnose as early as possible? We'll take in upper GI endoscopy or endoscopy in general as an example. So endoscopic diagnosis in the esophagus, finding this contrast is difficult. So we're trying to find contrast between, say, healthy and suspicious tissue, not necessarily diseased tissue, but suspicious tissue. Why is that difficult? Well, the standard of care, which is white light endoscopy, just simply replicates the human vision system. So the human vision system has not really evolved to be a good early stage cancer detection system, or has the ability to differentiate between different tissue types. So a good example of this is these images here taken from this second reference here. And the idea is that we're simply using a white light endoscope and we're looking in the esophagus of a patient. mean, to the endoscopist or somebody that is looking at this information, where are the suspicious regions that we were to pursue further? And by that, whether that is taking a section for pathology, whether that is even going further to examine kind of optically here, right? And what is shown in this paper is that it's very, very easy to miss, right? These regions here are the circled regions where a group of endoscopists have got together and said these are the regions of interest here. And the reason why it's difficult is that there are very subtle changes here. Right? So it's very easy to miss these kind of precursor lesions, dysplastic tissue or otherwise. And what is normally done because of this is that there are multiple staggered biopsies that are taken. So there's something called the Seattle protocol, and essentially what is done in a potentially suspicious area, there are these biopsies that are taken at very specific points in the esophagus. But as you can imagine, you are kind of undersampling, if you will, this region here. So it's very, very easy to miss regions that that are potentially suspicious. So, I mean, that's arguably not good enough. And can we do better? I mean, my background is physics here. Can we kind of look at some other ways to kind of alleviate this problem? So there's this idea of optical biomarkers, and the previous speaker, Caterina, spoke a lot about this, and it was an excellent talk, and there's a lot of extremely useful information there. But generally, we're throwing away loads of information, loads of the properties of light which we could potentially use. So, I mean, number one, optical imaging generally is a very useful modality or technique. It's rapid, non-ionizing, etc., etc. But there are lots of these properties that are associated with different constituents of the tissue. So, for example, having a look, but detecting, say, the phase profile of light, the polarization state or the depolarization of light, the spectral characteristics of the light that is either reflected, absorbed, or scattered from the tissue, etc., provides or can reveal very useful information about the tissue itself, whether that be endogenous absorbers or chromophores within the tissue, different depolarizing structures, say collagen. This information is lost. The standard of care, this is kind of got rid of, and we just look at the red, green, and blue channels of these very broad spectral bands. So could we potentially utilize this information? And the long view of this kind of research is, well, we want to, and as things are getting smaller and are becoming not necessarily more invasive, but the endoscopes are going into more and more places, let's say, could we integrate these different kind of optical imaging modalities into a single miniaturized system? So could we multiplex, this is the ideal and long-term view, could we multiplex these different properties in something small, in something, compact? Ideally, it would be cheap. It's not the end of the world at this stage, but also maintain the state of the art. So high resolution, red, green, and blue capture, and fast, right? This is clearly the long-term view. I'm going to focus particularly on spectral imaging here. And I know a number of people have already spoke about spectral imaging here, but just to recap, spectral imaging, typically what is it? So combining spatial and spectral information of some imaging system or some apparatus. Most people are very much familiar with a red, green, and blue or Bayer filter, which have these kind of three very broad spectral channels here. Everyone's smartphone in the audience will have a Bayer filter on their sensor, which can spectrally discriminate the light into these three channels. A lot of people are familiar with spectroscopy as well, where you have no spatial information and very, very detailed spectral information. Jumping across here to say hyperspectral, you have a ton of spectral information. So you have a near continuous spectral dimension and also very detailed spatial information. Multispectral, somewhere in the middle. So, whether you would have, say, 6 bands, 9 bands, 12 bands. I also might use bands and channels interchangeably here, which, if there are any mathematicians in the audience, I apologize in advance. But, you have relatively good spatial information, relatively okay spectral information, let's say. To implement something like this in the multispectral case, I'll talk about this a little bit later on. You typically use kind of snapshot. There are other ways, but you typically use kind of a snapshot multispectral sensor in which you say use this Bayer filter idea in which you have this mosaic on top of an image sensor, but instead of having just kind of these broad red, green, and blue pixels, how about now you have very narrow band filters atop of or integrated in this mosaic, and then you can demosaic the information here, and you can say reconstruct the very narrow band signals that are in the scene. So that's just kind of an overview. Maybe I should have had this slide earlier, but the spectral, and Caterina talked about this previously, these spectral signatures can provide very useful diagnostic information and can be used to reveal, say, and detect different biochemical changes due to disease development or otherwise within the tissue. And the different contrast agents that we could potentially look at, I mean, this is not extensive here, but the endogenous contrast agents, whether that is the the differential absorption spectra of oxy and deoxyhemoglobin, whether that be the exogenous ones, or if we to add a dye, a fluorescent dye, or there's a range of different contrast agents that we could potentially probe. And the beauty with spectral imaging as well, and some of these other modalities, is that it's wonderful if we can demonstrate them on a benchtop in a lab somewhere, but ideally we want to have an impact, and we want to help people, and we want to be able to implement this in a clinic or to actually be used. So the nice thing is that the spectral imaging configurations can typically be easily applied to chip on tip or baby scope configurations. And chip on tip typically refers, as the name suggests, to an image sensor right on the tip of the endoscope. Baby scope, there is a smaller scope that you can have as an insert into, say, a conventional approved endoscope. So the ability to translate a spectral imaging system into the esophagus or a colon trial, let's say, it's much easier with say spectral imaging than compared to some of the other systems. The Vision Lab generally, just as a small side note here, we kind of develop a range of different spectral imaging tools, and also the clinical trials as well. So one example is spectral endoscopy using kind of off-the-shelf components here. I'm not going to touch upon this too much, but it's just to highlight it if people are interested, that we also do or create systems which are just using off-the-shelf components and performed in clinical trials. Second example here is also looking at hyperspectral analysis of the tissue that is resectioned from the human esophagus in another clinical trial. I'm not going to touch upon them too much. It's just to highlight that also kind of goes on within the group. What I am going to talk about, and what I'm going to try and get to the meat of this now, is that from my view is that, well, hyperspectral imaging, it is very useful and it's excellent, but getting that information comes at a cost, right? And this highlight here is from this review article here, which is Surgical Spectral Imaging in this journal here. And this is just to show very common techniques for trying to obtain this hypercube's worth of information. And here are some typical approaches. And a lot of people are familiar with this kind of filter wheel approach, in which you have these, very narrow band filters that are in some filter wheel type arrangement. And then you just kind of rotate this filter wheel around as fast as possible. Well, I mean, that's typically slow. It's bulky, quite limited optical throughput. Normal hyperspectral systems that one would typically have, yes, you capture the complete hyper, but it's normally slow. And by slow, I typically mean if you have an endoscopist that is looking at someone's esophagus or colon, you ideally don't want to wait for that information to be captured. You don't want to wait a second for that to be captured. You don't want to wait a few seconds. If it needs to be very fast. It needs to be quite, the endoscopist needs to be familiar with the information that is coming out, not just in terms of having that concurrent with, say, white light imagery, but also in terms of the rate, the sampling rate as well. Oh, sorry, when I say sampling rate, I mean video rate, right? So the typical systems are quite slow, also quite bulky and quite expensive. The other approaches, which is kind of somewhere in the middle, are snapshot approaches. So snapshot multispectral imaging, which you have this kind of custom, well, not custom, is you have these multispectral filter arrays which are atop the sensors, and these are much cheaper. They're not smartphone level cheap. There's also a spatial spectral trade-off, and there are also some problems with it as well. But my point is going to be is that this is potentially quite an interesting solution here. right? And how about targeted images? Right? We talked about some, maybe there are very particular points in the spectra that we're looking at, the absorption spectra of some of the contrast agents. How about targeted spectral images? Right? Because, I mean, off-the-shelf sensors are, I mean, typically not application targeted. And by that, I mean, you know, if you have a particular application, like you wanted to look at the differential absorption spectrum, just using this as an example, when you're doing oximetry analysis, you ideally, you know, you don't necessarily want linearly spaced bands from, you know, from the blue to the near infrared, because there's a lot of redundant information there. You don't necessarily need all of that. You might only need some of the, you know, maybe one of the isobestic points. You might need one of the absorption max of one, the absorption max of the other, etc. And the point is that, you know, for each application, you typically want a custom, ideally want a custom set of bands, right? So, for example, the biomedical applications, whether it be endoscopy or otherwise, I mean, could we have biomedically targeted images? I mean, the short answer is yes, but the longer answer is, well, how deep are your pockets, right? Because if you talk to the manufacturers, if you're not going to order however many million of them, it's not really worth their while, right? And the process itself to make these things is also quite expensive, and it's quite complex to fabricate, especially for custom multispectral filter arrays. A lot of fabrication steps, and the processing is not necessarily straightforward. So what is the state of the art? And typically I'll kind of point to IMEC, and these will supply the image sensors to a range of different camera developers or manufacturers, let's say, and they'll just integrate their filters atop of their own sensors. And typically what you can buy, you know, you can clearly buy your red, green, and blue Bayer filter. You can also have a near infrared channel, and then you get the linearly spaced bands, right? So there's this kind of one size fits all approach in which, you know, you try to provide these to as many different customers as possible. And so what if they're not perfectly aligned with the bands of interest? And then we have a hyperspectral approach. But the unfortunate thing with that is that you have to scan. There's something that has to be scanned here in order to get the 2D spatial information of the scene. Yes, you get detailed hyperspectral, but something has to scan, which takes time. And also most bands are arguably redundant for the application of interest, say here, and it's also expensive. Right, so next slide here. So my background is more on the nanofabrication side, on nanophotonics and plasmonics, etc. How about nanophotonic-based approaches? Well, you know, we've done that in the past, and a lot of people do that. You know, the long story short, you will create arrays of very tiny metallic or high-index dielectric structures. The geometry and the refractive index of these structures interact very strongly with the incident light, or the electric field of light, typically. And by tailoring these properties, one can create, say, different transmission filters, different reflection filters, etc. Because this response or this resonance with these nanostructured arrays is very strongly dependent on the wavelength of light. So by tuning these, you can essentially create these, say, little filters, which are typically quite straightforward to fabricate, one lithographic step. But there are many, many downsides associated with them, right? And the common problems that we face is that, well, it's still expensive, right? You either need electron beam lithography to produce this, or you need, say, a deep UV stepper, or you need to go even down to the extreme UV route, right? Which is extremely expensive, right? So there's this scaling issue as well associated with that, and also the bandwidth. I've not talked about necessarily the full width half max or the actual spectral properties of these things, but compared to, you know, very narrow band filters that one would get on a hyperspectral image sensor or multispectral sensor, they're quite wide, right? And there are additional resonances associated with these structures that, you know, is not ideal, right? So are there any other approaches? Right, so what we've been trying to do and what we've kind of developed is, you know, trying to keep in mind that it needs to be cost effective for it to be adopted. We've tried to introduce this kind of cost effective kind of custom I always hate this when people don't do that, put in the acronyms here. I should have, so I apologize. Multispectral filter arrays using grayscale lithography. So this is just an overview of what we're trying to do and what we have been doing, is to use an approach which I'm going to describe in the next slide, where we convert this kind of grayscale exposure dose to different cavity heights on a surface. We do that on the wafer scale. We do that using electron beam lithography to start with to demonstrate the concept, and also do that on the wafer scale using photolithography. So very typical eye-line mask aligner based photolithography to create kind of custom arrays. And we also now do this on image sensors with a quite a large image sensor manufacturer. And then we also do the readout of this kind of custom spectral imaging. So that's just a summary of what I'm about to talk about. So the approach generally, what we're trying to do, or what we have been doing, is that we have our monochrome sensor here. So it's it has no spectral response aside from the silicon band gap that's in the actual pixels themselves. We want to put a custom multispectral filter array of our choosing that we can make that doesn't cost the world, and we can iterate very quickly atop of it, and we can perform some imaging to say, right, yeah, these are the bands we want, and for this particular application, right, that's it, we've got it, right, that's what we want to do. So what we've done then is that, well, in conventional clean room processing or photolithographic processing or lithographic processing in general, you typically take I'm sure there are many people in the audience that are familiar with this, so I kind of apologize for this, but we take a photoresist, so a polymer that's immersed in some solvent, spun on a wafer or some sample, and that polymer or that photoresist is sensitive to the incident energy of either electrons or photons. And the resist sensitivity curve, dependent on its tone, whether it's positive or negative, essentially looks something like this, right? In which, if you give, this is for a negative tone resist, if you give it a lot of energy here, a big exposure dose, then the resist, this is just normalized thickness of the resist. The resist remains, right? So it's there. If you don't give it anything at all, it's gone, right? So you have this kind of binary-like structure, which for most applications is fine. That's most of photolithography. You either want resist there as a sacrificial layer, or you don't want it there, right? But there is this other region in between, right, which is this kind of grayscale region in which people utilize for a range of different applications, whether it be diffractive optical elements or otherwise, in which if you give it a little bit of energy here, right, not everything's gone, but there is still something remaining, et cetera, et cetera, et cetera. Right, so if we do that atop of some mirror layer, whether that be an all dielectric stack, or to start with, say, a very thin or optically thin silver mirror here that is encapsulated, and we do this process, we can create these little cavities here. And I'm sure a lot of people are familiar with kind of Fabry-Perot-like structures. I think there was something in the quiz about this. That if you change your cavity height here, the center wavelength of operation of one of these cavities also shifts. Right? So what we can do then, we can structure these cavities with a grayscale exposure dose and change the center wavelength of transmission. So this is kind of what we do here, and the simple overview is that exposure dose controls the cavity thickness, right? So hence the center wavelength of the filter. So I mean, this is just simulation experimental, but this is just showing here in B that if you increase this is just highlighting the title really of the slide. If you increase the exposure dose, right, your transmission response of one of your cavities will increase, right, will increase. And then this is just from an atomic force micrograph here, just showing the cavity. I should have really plotted it the other way, but this is the cavity height as a function of the exposure dose here. But I'm a visual person. I like visuals here. So what this looks like. So this is kind of experimental results here. So if I apply exposure dose profile that looks something like this, this top image here is from an optical microscope in transmission. And then if I were to take that area and have a look under an atomic force microscope, it would look something like this. And then the same again here for little pixels here that are closer together. Now, what we can then start doing is that if we prove that concept, we can, we potentially have a simple yet effective, approach to facilitate custom multispectral filter arrays or spectral arrays in general, right? So, what we could do, what we have done is, well, we could do an RGB plus near infrared filter. That's wonderful, of course, and a lot of people do that. Also, we can do, say, a 4x3 filter here, do something like this, and also do some kind of crazier other structures, which, you know, there are a lot of well, that's obviously debatable. There are potentially applications available for, but there are a lot of interesting other structures that one could potentially think about that we could make. So that's with kind of electron beam lithography, which is still quite expensive and slow. But hang on a second, we want to do this on the wafer scale. So we do this as well. So we can now do this on the wafer scale, in which we do exactly the same system, exactly the same approach, sorry, in which we have multispectral filter arrays with very custom bands here, and we can do that, you know, on 3-4 inch wafers, etc. What we're also doing now, because I mean, trying to tie it back to the biomedical side here, is that, you know, we're trying to also have images here, right? Either we fix these multispectral filter arrays atop of our sensor, or we just fabricate directly atop of them, right? And that's easier said than done, but we have an agreement with quite a large manufacturer of image sensors, which they'll supply us they do supply us the raw sensors and the ceramic packages as well. So I will then fabricate atop of them custom multispectral filter arrays here. So an example here of 2 different sensors, or 3 here, but 2 different sensors, say, characterized here, the transmission response of 2 different ones. And, you know, without going into detail, you know, they're custom jobs here of different filter responses dependent on the sensor and the application here. So we take them, process, and then do the wire bonding in-house as well. And then it's just an AFM here of one of the equivalent areas, right? So I guess just, I mean, show that it's, I mean, working, I suppose, is that, you know, this is not the greatest demonstration in the world, but the point is that we're taking a custom multispectral sensor that we've kind of fabricated on in-house with a mosaic that has a transmission response like that, like this here. Could do very basic RGB reconstruction as well. The raw image would look something like this. And then we can also do, without going into too much, we also do having a look at, say, the spectral contrast between different channels here. And then we can start, you know, in the process of trying to scale this up, so we can start then applying it to particular biomedical applications with the goal that we want to do trials to begin with one of these sensors here. I appreciate the time here, so I'm gonna the last part here is that, well, what's next really? Well, this is just one thread of trying to integrate different, say, imaging, optical imaging modalities here. And when I'm using that with in particular with, say, spectral polarization, etc., not something like narrowband or fluorescence. You know, they can all be summarized with, you know, the illumination system, detection system, and what properties of light one is trying to look at. You know, trying to integrate all of these different properties into one system, right? So this is just one thread. So, you know, if we did have the potential to start with anyway for the spectral side, if we did have the potential for bespoke multispectral images and or multimodal one, what features would be the most, this is just the current thought process, kind of like what features are most desirable. And, you know, this is kind of a chicken and the egg question in some sense, and one may argue that, well, shouldn't you have asked that to begin with? And you can go round and round, but what can you produce? And you go round and round. All right, so whether that is narrowband spectrum, whether that's going into the near infrared or shortwave infrared, right? Does it have to be ridiculously low cost, right? In order to be adopted, does the cost of these things need to be equivalent to this to smartphone sensors, such that anyone can use them and it just becomes extremely cheap? I mean, something I didn't touch upon is that the chip-on-tip endoscopes now, the ideal, what's being sold are disposable endoscopes, so disposable chip-on-tip endoscopes, right? If you needed to throw away a £10,000 sensor each time you do it, well, no one's gonna buy it. No one's going to use it. Or is the main feature where it has to be simultaneous red, green, and blue white light imagery as well, such that the endoscopist or whoever is familiar with that information? And then how about polarization, phase contrast, etc.? All of these other kind of features here. So I'm just kind of leaving that open, is what we're trying to kind of move towards this kind of all-encompassing, say, multimodal imager, but particularly priority, like what is most important here, what is most important to incorporate. So in the interest of time here, you know, it's just some acknowledgements. So the PI of the group, so Professor Sarah Bondique, some of the people that have now moved on to other roles. So kind of George Gordon, who's over in Nottingham now, starting his own group. Travis Sawyer, who's also starting his own group in Arizona. Dale Jong-hee, who's also just about to start his own group, and some other kind of collaborators here. So thank you to the sponsors. So always happy to take questions, or please get in touch as well. Always happy to chat about a range of different things, research or otherwise. So thank you very much.
Thank you, Calum. The presentation was amazing. I love when figures are self-explaining and also there are little words and we could understand everything from just first sight. So I love seeing how it is possible to have a look at the synergy between also the speakers that we had today. In your presentation, I saw both like Cristian Manzoni's talk about hyperspectral imaging. Then I saw a possibility of implementing Gianmaria Calisesi technique with, I was thinking about Hadamard pattern for compressive sensing reconstruction of images. And this could be a great way to lowering the cost and the time of acquiring those images. And then I saw also some synergy between the Caterina's talk about scattering. What could be the role of scattering in your image reconstruction? How deep can your light go, and how well can we reconstruct those images? Even thinking about going a few millimeters deep inside the tissue. So what I was wondering.
No, it's tricky. It's funny. I was just about to comment that there are maybe too many interesting things to do and maybe not enough time to do them. But I would agree that it's a nice compared to some of the previous talks. There's nice kind of connections between them here, and there's enough of an overlap in each that you can kind of connect the dots, and you could see how, if you combine this and this, well, it could lead to this. One such area is compressive sensing, and it's one area that we're potentially interested in at this present moment. And on a personal note, I very much, Andy's talk at the start, I'm fascinated and very, very interested in kind of computational optics, and I was going to say everything associated with that. I do find it absolutely fascinating. So there are certain kind of routes that we're trying to pursue as well, and it's difficult because I think we try to kind of keep in mind that, well, in order for something to take off, and when I say take off, I mean used in a clinic kind of long term, or we want a manufacturer to integrate it, it has to do, typically anyway from the feedback, it has to do what the current systems do. So what's normally the response is that, well, if it takes even a second or half a second to produce some imagery, and then typically getting a 4K, very high resolution, red, green, and blue, lovely HD image from their system, 60 frames a second, whatever, 100 frames a second, right? It's not good enough to, if you did have something that is this, even a typical, whatever, regularization algorithm in compressive sensing takes, I don't know, a few seconds to do normally. I'm happy to be wrong, but normally a few seconds to do. So it's great from a research paper point of view, but from an application standpoint, it then becomes a little bit trickier. So I'm not giving any conclusion here. If anything, I'm just rambling on, but I'm just kind of thinking out loud here. So it's the challenge trying to bring together not only the biological side and the kind of the systems instrumentation side, but also the idea of what it has to do certain things in order for it to be adopted by a clinic or by some company or something like that. So it's kind of tricky trying to navigate that, and I don't have a good answer for most of this.
Yeah. I was just wondering, as you said, I think it's good to think about all these new application, and I see that there is much potential in other 1,000 directions, as you said. And I have a technical question about the filtering capability of of the device you showed us. So I've seen that you can filter different bands, but what are the bandwidths generally? I'm talking about full width at half maximum, approximately. I guess it depends on the wavelength, but just to have an idea.
Yeah, so with a with say a typical Fabry-Perot or thin film filter, the center wavelength controlled by the cavity thickness generally, and the bandwidth is typically controlled by the finesse of the cavity or how good the mirrors are. So we can control that. So I mean, the short answer to your question is about 20, 30 nanometers or so. Right? That's the very short answer to the question. The longer answer is that we can go narrower, we can go wider. It's dependent on the I was going to say the application, but technically dependent on the mirrors. So in the simplest case with silver, how thick those silver mirrors are. If it's the all dielectric case, well, how many layers are you going to have with this high-low alternating structure? So yeah, that was the short answer. Tens of nanometers or so, and the longer answer, it can be controlled.
Yeah, and I guess that then there is the trade-off between being cost-effective and high feature device. So if you want it to be used, it must be cost-effective.
Well, exactly. Yeah, exactly. Yeah. So it's yeah, I could ramble about this for a while. So
yeah. Okay, thank you very much.
Thank you.
I guess we are at the close to the end of the event. I leave the floor to Salva, who was one of the person that put the most energy inside this event, and so I thank Salva a lot for his energy and his commitment.
Well, thank you very much. Yes, just few words before we close up this meeting, this first edition of Eumos, and I would like to thank all the speakers for their time and their willingness to participate on the event. Without them, this event wouldn't be possible, and we wouldn't have heard of such great technologies as Michael highlighted, how convoluted they are and the many possibilities in terms of future research and future applications for different fields, from gas sensing to healthcare applications. I would like to also thank, in name of Cisma, the 3 other societies we collaborated with to organize this. So the people from OSA Milan and Young Minds, Scope from Strathclyde University, and FISUA from Aveiro, and of course the Scottish Universities Physics Alliance, SUPA, and the Centre in Doctoral Training in Intelligent and Sensing Measurements for supporting us economically and making able to have Gather Town, that I think that everyone love it, and to run the quiz and the poster session. Also from the groups that made this event possible, I would like to do a little bit of promotion about future events. So from the people in Milan, they're organizing a Women in Science event. You can have the information by having a screenshot or a photo on the QR code, or just going through any of the links on the screen. So I'll wait a few seconds so you can have that screenshot. Don't miss it. You know, from Cisma, we loved to work with them since minute 1, and they're doing really cool stuff. So please don't miss that one. Also from the Strathclyde people, they're having tomorrow a Ask Me Anything event. That sounds interesting. You know, one of the questions I will ask is if there is any other answer to what is the ultimate question of life, the universe, and everything. Hopefully it's not 42, but you know, that's an answer for tomorrow. The same in here. Follow the links or take a screenshot to the QR code on your screen. Then we have people from FISUA, Beiro. They're also organizing quite a few things along the year. Also have a look to the links in there. And finally, as Cisma, we have more than a few webinars, monthly webinars from today until July. Then we have a symposium for sustainable future human in September, and the third edition of the future of ISM this year, wearable devices, in November to be concrete, but yeah, we will see that soon. And finally, we have something new, something that we recently launched, a podcast that we're organizing. The first episode was uploaded yesterday, so go to Spotify and it's in there for free for you to listen. Believe me, it's quite worth it, and it's quite funny, so don't miss it. Okay, the quiz winner is Big Ollie Glasgow, so please, Big Ollie Glasgow, get in touch and we will manage to get your prize in your house. Thank you very much for participating, and well done. And the poster winners, because we had a draw in the pool that we had, so we decided to split the prize. Are Lorenzo I'm going to say this wrong, so I'm very sorry. Frabasil and Pietro Levoni. We will also get in touch with you to get the prize for you. Again, thank you everyone for being here, and I hope to see you all soon. Have a nice day. Bye-bye.